Good morning, everybody. We are starting the third Tuesday and the last session, the last two sessions of our ACDIS Cancer Spring Symposium. Today we're going to talk about system biology, immuno-oncology, and nanotechnology. I will be in charge of system biology and immuno-oncology section. So we have 4 speakers. Each one will have 25 minutes, and questions would be at the end. We will have a roundtable discussion at the very end of the session. So this session is mixed between system biology, the first 2 speakers, and immuno-oncology, the third and fourth speakers. So it's a mix about specific topics in cancer that are absolutely relevant and probably is a way to move in the future. So with that, I would like to introduce our first speaker, who is Dr. Hermes Fernandez, who obtained his PhD from University of Santiago de Compostela in Spain in 2003. And then he did his postdoc in toxogenomics at the University of Antwerp in Belgium with Dr. Bart Naus. In 2004, he moved back to Córdoba, Argentina, to become an independent researcher for the National Council of Scientific and Technical Investigation of Argentina. Since then, he leads the bioscience data mining group of the Research Center in Immunology and Infectious Disease of CONICET, Argentina. His line of research is the development of statistical computational techniques for the search for information in biomedical sciences. Dr. Fernandez?
¿Me escuchan? Can you hear me?
Perfect.
So I can share, yes, I can share now. Okay. Are you seeing?
Yes.
Okay. So thank you, Alejandro, for the introduction. Thank you, everybody, for, especially the ACDIS people, for organizing this wonderful meeting. I'm going to talk about some applications of big data analytics in cancer. In particular, I'm going to show you an example that we've been working in the last few months in applying some FDA-approved molecular signature that was originally developed for a particular cancer type in a cross-cancer application. So, as you may know, the first decade of the 21st century was characterized by the development of thousands of molecular signatures. However, only a few of them reached the market and were FDA approved, even though some of them are still in this process. In the same period of time, the Cancer Genome Atlas began with, among several goals, I would like to recall 2 of them. The first one is that one of these goals was if molecular signature can be used in therapeutic decision making, something that was already proved. And the next goal that I would like to recall is that they think if some of these predictive expression-based signature can be used as a tumor agnostic biomarkers. Tumor agnostic biomarker is a hot topic today. We can mention, for instance, the tumor mutation burden that is currently used for splitting the subjects in immunotherapy, in cancer immunotherapy. And taking this into account, and also the chance that the TCGA project give us to afford or to face the all the hallmarks of cancers in several subjects, many authors try to look for commonalities between one cancer type, another cancer type, using subtypes defined in one of them. In particular, I can mention that in this case, the SAO group tried to apply PAM50 across many tumors, but for some reason, he kicked off some of the tissue types, for instance, the not epithelial ones, and also those that express HER2. However, HER2 and subtype has been found, or some commonalities has been found in other tissue types beyond breast cancers. Another thing that we observe that is if you apply a particular molecular signature on an expression profile, basically all the biomarker strategies will give you a straightforward answer. So they will say this is class A, class B, or class C without any doubt. So they never say, I'm not quite sure about my assignation. However, we prove that if the data are not appropriately processed, some result may be misleading. So the system can change the decision regarding the processing process. And in addition to that, we also observe that some of the patients in breast cancer have been assigned with very little information support. For that reason, we developed a few years ago a method to provide statistical confidence to the assignation, to the PAM50 assignation. For instance, if the subject cannot be confidently assigned, we observe that they don't have difference in the outcome profile regarding their subtype. And by using this approach, we observe that we can split the subject before, much better than using just the PAM50 algorithm. So with this in mind, we applied or reapplied this process to the whole TCGA cohort. And we found that, as previously shown, in the breast cancer case, approximately 26% of the samples cannot be accurately or confidently assigned. And for the rest of the cancer, this proportion is much higher, which tell us that you can't assign a cancer, subject to a specific subtype without certain confidence, because maybe this subject has been assigned randomly. So we decided to kick off these subjects from our study and put our focus on those subjects that has been confidently assigned, because now we want to see if the molecular characteristics can transcend tissues. We decided to do this approach by using a functional approach, because we already saw that if we use gene expression, the overlap in the analysis is not too high. So by a functional approach, the overlap when you have a particular disease across tumors and across platforms, it's much higher. So we use two approaches. One is gene set variation analysis, which provides us a score, an enrichment score for each subject and for each of the gene sets taken into account. And we use the massive and integrated gene set analysis, which is a tool that we developed and published last year that allow us to compare different contrasts nested into different tissue types and provide us a global overview of the results across different cohorts or platforms that we include in the experiment. By doing this, we can see that, for instance, basically the main difference between the different subtypes are associated to luminal A subtype against the rest. When we compare the other subtypes, we don't see pretty much differences from a functional perspective. So basically, MIXA tell us that there are not 4 groups, there are basically 2 groups. This observation can also be supported by using dimensional reduction technique that we can see, for instance, that basically the subjects group together regarding the luminal A case, and the rest of the samples are mixed in a different cloud. Let's say this is breast cancer, which is the only one who tried to show or seem to show the differences between the 4 groups, but this is not quite clear. So this suggests that across, in a tumor agnostic way, we don't have basically 4 groups. We basically have 2 main groups. We call them class 1, which is basically representing luminal A case, and class 2, which is basically representing the rest of the breast cancer subtypes. So this is our approach, is that if we are testing a subject, so if we can assign it confidently, then we can classify it in 2 classes, just using the original PAM50 classification and regrouping in this way. Or if this assignation cannot be confidently done, so this subject may follow the traditional path in the clinic. So in order to compare now the differences, if we can find some particular differences that identify these 2 subtypes, we split all the population in this class 1 and class 2. And we evaluate the difference from a functional point of view and also from a gene expression point of view in order to see if we can find coherent patterns across the tumor types. And this was the case, for instance, for the gene set analysis. We can see that where we can find 28 gene sets that were coherently expressed across tumors, differentiating class 1 against class 2. When we use gene expression approach, we were able to find 95 genes that were coherently expressed across all the tumors, differentiating these 2 classes. Here we split the population in breast cancer, other epithelial and non-epithelial ones, in order to see that this the PAM50 was developed for breast cancer, and in order to see that this approach holds for the epithelial one that was previously used in other projects and in the non-epithelial ones. So now we wonder which are the main difference driving these differences. And we found that basically functional pathways related to cell cycle, tumor progression, and DNA damage response are the main topics talking about the difference between these 2 main classes. So in order to validate in some extent these findings, so we look for other molecular signatures that point directly to some specific pathways in cancer that were also evaluated in different tumor types in the sense following in some extent the hallmarks of cancer. And we were able to validate them because our molecular signature, or our proposal, which is important to remember that this is an FDA-approved molecular signature, reflects the differences already seen by functional approach. So we use a proliferation score, a centrosome amplification score, which is talking about spindle checkpoint pathway, a very common pathway, which is TP53, and the retinoblastoma tumor suppressor pathway, which is a molecular signature that talks about or provides us information about DNA damage and apoptosis pathway. We also see that these two main classes provide survival information. We found 8 tumors from the whole TCGA cohort that were significant differences between the outcome between class 1 and class 2, being worst class 2. And among them, 5 were very significant from a statistical point of view because this assignation cannot be achieved by random signatures. The next question that we made was that if we found 95 genes that were very coherently expressed across all the tumors, which means in some extent that it's a pattern profile. So if there exists some possibility that these genes could be driven by some transcription factors that are usually associated as master regulators. So in our list of 95 genes, we found 9 transcription factors that were overrepresented, or overexpressed in the class 2, which is the most proliferative and the more differentiated class. And among them, we found 2 that target 87 out of the 95 genes present in the gene list. And these genes are currently under research as drug targets and also as a master regulator of cancer, which is FOXM1 and E2F1. So this suggests that this process can be driven by only 2 genes. So maybe these genes can be associated as a hub genes that are good targets or research targets at least for controlling this process. In addition, we also want to see if our observations are beyond TCGA. So in order to see if this process is general from or can be used in other in a broader way using different platforms and or other cohorts from different countries. So we choose 6 gene expression cohorts from the gene expression omnibus. We pay special attention on those cohorts that that have enough patients and also have survival information, and also use the 1,000 samples tumor cell lines from the genomics of fractal sensitivity in cancer, which is a project to test different compounds on cell lines. So we applied the same process and we tried to see, and by using the 28 gene sets and also the 95 gene sets and also this molecular signature that pointing specifically some pathways in the sample, to see if these patterns can also be seen in a different platforms and in a different type of biopsies. And we found exactly the same pattern across these 6 breast cancers, and 6 tissue types, and also in the 1,000 molecular cell lines that represent basically all the tumors in the TCGA. So we see this from a functional point of view and also from a gene expression point of view, and also by using these other molecular signatures not FDA approved. So then we also wonder if it would be possible to associate some compounds, some drug that can target the genes present in this very coherent pattern across tumor types. So we did 2 approaches here. One by using the Enrichart tool that in their data in its database has different gene sets associated to drugs, and by feeding the tool with this gene list, we found several drugs compounds that may possibly target these cheats. In addition, then we use the inhibitory concentration 50 score from the genomics of drug sensitivity in cancer, and we found several drugs, several compounds that were that present different IC scores regarding the classification schema that we propose here. So we saw that most of the compounds involve or are targeting apoptosis regulation, cell cycle, and different other pathways related to cell cycle proliferation and DNA damage response. In addition, we also evaluate the immune content of these two classes, basically because the immune system is currently under strong research because of their success as an anti-cancer therapy in different tumors and in different kind of subjects. So in order to query that, we developed a tool called Mixture that was recently published. It's in process of publication. That by using a particular molecular signature, in our case, we use the molecular signature developed by Newman that provide us the information about 22 immune cells that can be present in the tumor microenvironment. So by using this, we interrogate the tumor microenvironment, and we found interesting things relating that showing a stronger interaction between the proposed classes, which is basically a proliferation class and a low proliferation and a high proliferation class, or a high differentiated class, and its tumor microenvironment. For instance, we can mention that the macrophages M1 cell type, which is basically high levels are basically associated as an anti-tumor cell, we can see that it shows interaction regarding the molecular profile of the tumor, where here a low level of M1 macrophages seems to be a good prognosis in class 1, but high levels are related to bad prognosis in class 2. So we think that this kind of analysis deserve further investigation. So as a take-home message, I can say that multi-omic data platforms are crucial for us in order to evaluate our findings in a broad overview across different tumors. So storage and sharing of data is crucial. Unfortunately, Latin America data is very scarce. In most of the databases, we are severely underrepresented. That's why projects like the Latin American Cancer Research Network is very important, and we need to favor these kind of projects. We demonstrate that the confident assignation is an important feature for accurate patient class therapy assignation. So any biomarker should also be joined with this kind of process in order to provide confident assignations. Molecular signature repositioning seems to be feasible and also provides meaningful biological information across tumor types. Molecular and immunological patient stratification may help therapy guidance, and is a hot topic today because there are several clinical trials combining chemotherapy that is guided it's guided by molecular profiles, and immunotherapy, which is guided by immunological profiles. So this is a hot topic today. And also testing FDA-approved biomarkers in a pan-cancer approach may allow discovering new potential use of this biomarker in other tumors that were not originally designed for, and faster the bedside translation. As you may know, today the translation from bench to bedside may take 10 or more years to reach the patient. So I want to thank all my collaborators, especially Darío Rocha, who is my PhD student, he did mostly all the work, and all the rest are our advisors in different topics: Dr. Ayera, Dr. Girotti, Dr. Soria, Dr. Garcia, Dra. Aldana Montoro, y Laura Prato, who help us in different topics of the development of the project that I already shown to you. So thank you very much for your attention.
Thank you, Elmer. As I mentioned in the chat, we'll take the questions at the very end of the session. So you can put your questions in the chat, and we're collecting those questions. So at the very end, we will have a roundtable discussion. So now we'll move on to the next speaker, who is Dr. Vinicius Maracaja Coutinho. Dr. Maracaja Coutinho is a PhD in bioinformatics from the University of Sao Paulo. He moved to Chile to create a company, bioinformatics, Beagle in bioinformatics, with support of Corfo and Startup Chile programs. In parallel, he started his research group in 2013 at Universidad Mayor, and then he moved to Universidad de Chile in 2018, when he also took the position of as an associated researcher of the Advanced Cancer of Center for Chronic Diseases. Dr. Vinicius is also a permanent faculty of the PhD program on bioinformatics, multidisciplinary environment of the Federal University of Rio Grande do Norte, Brazil, and visiting researcher in Dr. Tony Kousorides' group at the Gurdon Institute, University of Cambridge. With that, I would like to introduce, welcome Dr. Maracaja, Vinicius Maracaja. Oh, that's your presentation. Okay.
Good morning, everyone. First of all, thank you very much.
Press play.
Please. Very much the organizing committee for the invitation to be here to show, and the opportunity to show a little bit of what we are doing with bioinformatics, lung noncognianase and cancer. I'd like to start my talk with this sentence extracted from a manuscript from 1991 from Walter Gilbert, in which he said that to use the fluid of knowledge which will pour across the computer networks of the world, biologists not only must become computer literate, but also change their approach to the problem of understanding life. And since the first publication of the human genome, the cost to sequence a genome decreased drastically. The first publication costed about $3 billion, and this year I was able to sequence my entire genome for only $299. And this decrease in the cost is generating a life science big data revolution. And novel initiatives like the Cancer Genome Atlas is participating and generating a huge amount of data that are there able to be explored by different scientists from the world. For example, the Human Genome Atlas, the Cancer Genome Atlas, they generated 7 different data types for proteomics, metabolomics, genomics, transcriptomics for 11,000 patients for 33 different tumor types. And one example of this kind of data, of the database that integrated public data sets, is the Sequence Read Archive, which integrated more than 2 million experiments. Another example was the Gene Expression Omnibus database that integrated majority data from microarray experiments with more than 2 million samples specifically for human. And these data, they are there waiting to be explored. And the main bottleneck is to understand this data in a way that you can organize it in a standardized way, allowing the development of novel tools and databases that can be used and applied to obtain novel insights for relevant biological questions. These are the three main topics of the Laboratory of Integrative Bioinformatics, the laboratory that I lead at the University of Chile. In this talk, I will talk briefly about different projects that we developed along my career. I will split it in two main parts. The first part is the two works that opened my mind to the powerful of integrating public data in order to obtain and retrieve relevant biological information for long-non-code RNAs. And the other three are three works that are, one is already published, the other two are ongoing work that I started all them after I started my lab here in Chile. At least 76% of the human genome is transcribed in a sea of non-coding RNAs, with only 2 to 3% of the transcripts of the human genome being transcribed, and the remaining being transcribed specifically into two main classes of non-coding RNA, the small non-coding RNAs and the long non-coding RNAs. It's known that these non-coding RNAs, they are already described to be participating in several key biological processes in the cell, and that the regulation of these transcripts can lead to different sets of human diseases like cancer, neurodegenerative diseases, and others. When I started my career, the laboratory that I was working, they were participating in the global initiative to obtain ESTs for the human cancer. And at that time, they saw that several thousands of these ESTs, they did not map it to the protein coding genes, and they were actually mapping into intergenic and intronic genomes of the genomes. And that called the attention of the group in order to explore more deeply these intronic and non-coding RNAs. And that was the first opportunity that I have to use public data sets in order to obtain relevant information for characterizing long-coding RNAs. At that opportunity, the group started to integrate data sets from the DB-EST, a public available database for ESTs from the NCBI, in order to retrieve these ESTs and reanalyze them in order to obtain and clusterize all these transcripts into these ESTs in order to generate sets of potentially novel long non-coding RNAs originally being transcribed from intronic regions of the human genome. And after generating these sets of potential intronic transcripts, the group developed an in-house oligoarray platform composed by intronic and exonic transcripts with probes that target for these intronic non-coding RNAs and their particular host gene, the genes that host the intronic region in which these transcripts are transcribed. And these microarrays, they were applied in order to explore the transcriptional coding and intronic and protein coding expression patterns of different cancer types in the Brazilian population. And the first work that I would like to show you, we integrated the information of these microarrays with some public available data in order to characterize some intronic and non-coding RNAs in pancreatic cancer. In this work, we firstly found 43 long non-coding RNAs that were differentially expressed between primary tumors and non-tumoral samples. Next, more interestingly, we found 134 long non-coding RNAs that were differentially expressed between primary tumors and metastases. And that was one of the first times that long non-coding RNAs were shown to be to perform different expression patterns in metastases and primary tumors. We next showed that these non-coding RNAs, similar to protein coding genes, they are regulated by stun modifications. At this time, we showed that they are regulated by methylation by the integration of public available data generated by the ENCODE project. Also, by analyzing the expression patterns of these non-coding RNAs, we showed that they are actual real independent transcripts. They are not a transcriptional noise of the primary RNA of the host gene. We showed that by analyzing the expression patterns of these non-coding RNAs and comparing the expression patterns with the expression of these protein-coding genes. For example, we found some genes that they were differentially expressed, the protein code, the long non-coding RNAs, they were differentially expressed. Meanwhile, the coding genes, they were not dysregulated in the condition that we are exploring. And that was validated by qPCR. Next, we integrated public available data for conservation from the UCSC genome browser in order to show that these transcripts, they possess some particular motifs that are more conserved than randomly selected genomic regions. Based on this data available in this database, we showed that these transcripts possess these motifs that are more conserved between primates, placentals, and vertebrates than randomly selected regions of the genome. In another work, we used, again, these arrays and integrated public data in order to characterize intronic non-coding RNAs in renal cell carcinoma. We found in this work 48 long non-coding RNAs differentially expressed in renal clear cell carcinoma, and at this opportunity we used it for the first time co-expression model analysis as a tool for assigning to obtain functional assignations for these non-coding RNAs, functional associations for these non-coding RNAs, in a way that we can find modules or networks of transcripts that are systematically co-expressed. And we, for example, here in 19, we have a model that is enriched with long non-coding RNAs, and we found that these non-coding RNAs, they are systematically co-expressed with protein coding genes. And these protein coding genes, they are they are enriched, and they are normally participating in protein ubiquitination, polyubiquitination. So we can associate that these non-coding RNAs could be potentially participating in this biological process. So we integrated again RNA-seq public data from the ENCODE project in order to obtain an expression, to obtain the correlations of the expression and the co-expression patterns between protein coding genes and these intronic non-coding RNAs. And we found that these non-coding RNAs, we were able to associate these long non-coding RNAs with the gene ontology terms that these protein coding genes belong. So we are able to associate, for example, some types of non-coding RNAs with particular biological process in the cell. It was our first time, the first time that we were able to obtain at least some insights of the biological process that these non-coding RNAs were participating. And also we are able to integrate again public data from UCSC genome browser and from the ENCODE project in order to show that these non-coding RNAs, similarly to protein coding genes, they are regulated by PO2, by different promoters, and also by other types of histone modification, like acetylation and other types of methylations. We also show that these non-coding RNAs, again, they possess some particular motifs that are more conserved than randomly selected regions of the genomes, as we can see here. And we also show that these non-coding RNAs, as these RNA-Z data set here, they possess much more conserved secondary structure motifs than randomly selected regions of the genome. And this work motivated us, this co-expression analysis, to develop a particular tool called SemiTool, which is a bioconductor package for and a web server for performing comprehensive modular co-expression analysis. This is a work in collaboration with the group of Helder Nakaya from the University of São Paulo, and we developed two tools which can be used by people with a computational background through the bioconductor package, or for tools without programming skills, they can use the web server version of the tool. This work I started to perform when I moved to Chile, and I was already with my lab established, and this is a collaboration with the group of Tony Kozarides, and we described a novel type, two novel subtypes or subclasses of long non-coding RNAs. In this work at this time point, we already know that long non-coding RNAs, most of them, they can be transcribed and they recruit chromatin modifying enzymes in order to regulate the transcription of the neighborhood gene. I think these into account, we try to see the sets of non-coding RNAs that are conserved. They have the pair of a coding gene of a long non-coding RNAs conserved between human and mouse. We took all the protein coding genes in human that possess an autologous genes in mouse, and we used RNA-seq data from ENCODE in order to identify sets of long non-coding RNAs in both genomes. Next, we tried to see to select and filter the long non-coding RNAs that possess a promoter region that's conserved between human and mouse, in a way that we have a set of protein coding genes that are orthologous between both organisms, and they possess a long non-coding RNAs in their vicinity that possess a promoter region that is conserved between both organisms. We used all these pipeline in order to identify a set of 1,700 pcRNAs, which are these non-coding RNAs conserved in both genomes. We saw that these non-coding RNAs, they are located nearby genes related to development, as we can see here, cell differentiation, morphogenesis, and gastrulation. We saw also that these non-coding RNAs, they possess a conserved tissue-specific expression patterns between human and mouse tissues and cell lines. And more interestingly, we found that these non-coding RNAs, again, by integrating public data related to CTCF loop, the loop complex CTCF, which is a complex that regulates the topological formation of DNA. And we found that these non-coding RNAs, they are located, a subset of these non-coding RNAs, they are located by overlapping a motif of this loop. And we saw that these non-coding RNA, they possess a consensus motif that's quite similar to the consensus motif of the enhancer region located in the other side of the loop. And we believe that these non-coding RNA, they regulate the topological conformation of DNA by interacting with this region. And these non-coding RNAs, they do not occur by chance. This is something that we found that's enriched specifically in a subset of pcRNAs, and we call these non-coding RNAs as TAP RNAs, which means for topological anchor point RNAs. Since these non-coding RNAs, they are normally located nearby genes related to development, we believe, we asked if they could be associated with cancer. We retrieved several experiments for microarray microarray experiments available in GAO, and we found that 203 long non-coding RNAs, pcRNAs, they are directly associated with several cancer types that we can see here. And we also integrated data from the Cancer Genome Atlas and found that 403 pcRNAs, they are also associated with different cancer types available in TCGA database. Next, I will talk about a project an ongoing project that we are developing in collaboration with Paulo Amaral from the University of Cambridge. Then this work is led by Victor Aliaga Tobar from my group. And in this work, we are trying to see if long non-coding RNA, they could be regulated by RNA-modifying enzymes in cancer. For that, RNA modifications and epitranscriptomics, they are a new trend in RNA biology research. So far, we already know more than 300 RNA-modifying enzymes, with 77 of them described in human. However, only 140 modifications have been 140 modifications were described in tRNAs and ribosomal RNAs, with less than 10 in messenger RNAs and other types of non-coding RNAs. In this work, we integrated data sets from TCGA again, and we filtered the cancer types that we could retrieve at least 10 samples for normal tissue or primary tissue. Here you can see all the cancer types that we integrated. We were able to quantify the expression of all the coding transcripts, their RNA-modifying enzymes and non-coding RNAs for all cancer types. And we retrieved the sets of differential expressed coding genes RNA-modifying enzymes, and long non-coding RNAs. And in this analysis, we're able to see, for example, 10 enzymes that are highly associated with several cancer types. For example, here we have these 10 enzymes that are always systematically differentially expressed in at least 10 cancer types. And some interesting data also is that there are some cancer types that possess several RNA-modifying enzymes systematically differentially expressed. For example, esophageal carcinoma possess 29 RNA-modifying enzymes that are always differentially expressed in 29 RNA-modifying enzymes that are differentially expressed in only this cancer type. As I mentioned before, we were able also to obtain the sets of long non-coding RNA differentially expressed in all the 16 cancer types. As expected, we saw that these non-coding RNAs, they are more cancer specific. It's expected because non-coding RNAs, they possess a much more tissue specific expression pattern compared to protein coding genes. And next, we performed some co-expression analysis by obtaining the correlation of expression between long non-coding RNA and the transcript isoforms for all the RNA modifying enzymes, obtain the sets of transcripts and long non-coding RNAs and RNA modifying enzymes that are positively correlated in red and the negatively correlated in blue. The next steps of this work, we are performing now some co-expression model analysis in order to obtain modules of genes, modules of transcripts that we have. The idea is to find a model that we have a particular long non-coding RNA that is differentially expressed, and in that model we have a particular RNA modifying enzyme that's differentially expressed, and we are integrated in-house and public, in-house developed, in-house generated and public data sets from different epitranscriptome sequencing data in a way that we are eager to find some, if the particular enzyme and long non-coding RNA that we find here, they possess a particular modification in the cancer or in the normal tissue. And this will help us to filter some candidates to be functionally validated experimentally. The last work that I would like to talk, this is a work developed by Adolfo Rojas and Alan Peñalosa in collaboration with Michel Naslavsky from the University of São Paulo and Ignacio Wichmann from the Catholic University of Chile. In this work, we are trying to describe the competing endogenous RNA networks in breast cancer by the integration of public data. Competing endogenous RNAs, this is a mechanism in which a particular non-coding RNA, they act sponging and sequestering microRNAs, avoiding these microRNAs to target the original target mRNA in a way that this mRNA keep activated and being able to generate a particular, the protein and translating the protein that it's transcribed for. And these mechanisms already know and has been described in several different types of human diseases, including different cancers and cardiovascular disease. And in this work, in a very beautiful work by Alan Peñalosa, we generated a reference set of human microRNA mRNA interaction network with more than 580,000 interactions, composed by 4 different databases, including some computational predicted interactions in which we selected only 2 interactions predicted by the 3 databases, together with some interactions that were experimentally validated. He generated another reference database specifically for long non-coding RNAs and microRNAs interactions. He integrated data available in 2 different databases, which were all them experimentally validated interactions, and generated a total of 67,209 experimentally validated interactions. And he integrated both databases in order to obtain a human global long non-coding RNA, microRNA, mRNA interaction, which is composed by 105 more than 105 million interactions, composed by more than 7,000 long-non-code RNAs, more than 900 microRNAs, and more than 19,000 mRNAs. And we integrated data from TCGA specifically for breast cancer. We obtained the sets of long-non-code RNAs, microRNAs, and coding genes that are differentially expressed in this particular cancer type, in order to cross-reference these data and these their expression pattern in order to obtain a complete endogenous RNA network specifically for breast cancer. We filtered this network in order to obtain intermediate network composed by specifically long-non-code RNAs that were differentially expressed. We obtained a total of more than 162,000 interactions of 72 long-non-code RNAs differentially expressed in breast cancer, 85 microRNAs, and 1,128 protein coding genes. And these protein coding genes, they are enriched in some biological process related to breast cancer, like multicellular organ development, blood vessel morphogenesis, hormone stimulus response, and cell cycle process. As a next step, we are working on generating co-expression model analysis. We are working on identifying some subtype specific complete endogenous RNA networks according to different breast cancer subtypes. And we are going to apply different computational approach in order to select some particular candidates and generate a small number of candidates to be experimentally validated. As a take-home message, I'd like to show you what this text from the same manuscript from Walter Gilbert that I showed on the beginning, where he said that the new paradigm now emerging is that all the genes that will be known in the sense of being resident in database available electronically, and that the starting point of a biological investigation will be theoretical. Sorry. An individual scientist will begin with a theoretical conjecture, only then turning to experiment to follow or test that hypothesis. And this is something that he predicted in 1991, is already a reality. And as I show you, all the work that I showed here, it was only possible because of the integration of all these huge amount of data publicly available. This work was only possible because of this amazing team and the funding for the national scientific agencies. Thank you very much.
Thanks, Minister, for this wonderful presentation. Really amazing. Great. So, as I mentioned before, you can leave the questions in the chat, so we'll collect all of them to have a roundtable discussion by the very end of the session. So with that, I would like to move to the next speaker, who's Dr. Álvaro Yatzer, who studied biochemistry and PhD in biomedical sciences, PhD in biomedical sciences at the University of Chile, obtaining his degree in 2007. Then he did his postdoctoral training at the Cancer Center Karolinska Institute in Sweden, working on T-cell-based cancer vaccines with Dr. Rolf Kissling. In 2010, Dr. Yatzer moved back to Chile to start his career as an independent scientist at Fundación Ciencia Vida. Since then, Dr. Yatzer has developed his research focusing on the long-lasting T-cell mediated anti-tumor immunity. His current projects aim to decipher to clarify the crosstalk between CD8 T cells and dendritic cells in the tumor microenvironment, and to dissect the role of tissue-resident memory cells in human solid cancers. Dr. Yatzer?
Can you listen?
Yes, perfect.
Yeah. Thank you very much for the invitation and the presentation. So, as introduction, I will let you know how the anti-tumor T-cell response are initiated. So, when the tumor is developed, dendritic cells, which are the sentinels of our immune system, uptake antigens in the tumor site, and then they migrate to the lymph node where they present this antigen to CD8 T cells and CD4 T cells upon 3 different signals. Okay, so the specificity is given by the antigen, and they need also costimulatory signals and cytokines. So all the antigen-specific T cells, they clonally expand and differentiate into effector T cells. So they acquire the ability to migrate to peripheral lymph nodes, non-lymphoid tissues, and also to do the cytotoxic activity by recognizing the antigens in the surface of the tumor cell. So if we look this same phenomenon, but in the time, we can see that at the beginning, only a few cells are antigen specific. And then after the priming, these cells clonally expand and become the effector T cells. But after a while, most of these cells are eliminated, and only the memory precursors become the memory T cells that can last long. So among the memory T cells, we have 2 different subsets. So we have the circulating T cells and the tissue resident memory T cells. So these cells are very important for infection disease and also for cancer cells. So what is important about these cells? These cells, they establish in barrier and non-barrier tissues, okay? They're long-lived and they acquire a distinct residency signature, characterized by these 2 important molecules, CD69 and CD103. This molecule allows these cells, for instance, to interact with the epithelial cells expressing E-cadherin. And this is very important for the avoiding the recirculation of these cells, and they become permanently established in tissues. So what are the key features of these cells? They have a potent effector function. They produce large amount of gamma interferon, TNF-alpha, granzyme B, and other cytokines that are really important to do the cytotoxic activity, and also to trigger an innate-like alert state in tissues, which is very important to communicate with other cells. So these cells, they can recruit and activate dendritic cells, NK cells, T cells, and B cells. So for instance, here you can see that gamma interferon is really important for the recruitment of other CD8 T cells, and TNF-alpha is important for the maturation of dendritic cells. But all of these have been shown mainly in infection disease. So in tumors, recently, it's becoming evident that these cells, TRM cells, are orchestrators of anti-tumor immunity. So we have shown, along with other groups, that these cells are really important in eliminating melanoma cells in the skin. But by doing so, these cells can also crosstalk with dendritic cells to enhance anti-tumor immunity. So we have shown in this paper that these cells can communicate with dendritic cells to produce more anti-tumor CD8 T cells. And these cells can now come and eliminate more efficiently the tumors, the primary tumor, but also they can go to distant site and eliminate metastases. So all these studies have been shown for us and for other groups in mouse models. But we have also looked this into human samples. For instance, we have analyzed the database available in the Cancer Genome Atlas, when we can observe a similar communication between activated dendritic cells and TRM and cells. So this is a gene signature analysis when you can see a good correlation between the enrichment of TRM cells and the activated dendritic cells. As a control, we can look that there's no such association with immature dendritic cells. We have also shown that in fresh samples from breast cancer tumors, that the TRM cells can associate with other different DC subsets, different types of dendritic cells, like DC3 or DC1, okay? And they can even make dendritic cells to mature in the, and migrate to the lymph nodes. So this is all we have observed, this crosstalk between TRM cells and dendritic cells in human tumors. But our next question was to address if these cells have an impact on the survival of patients. So we did also a gene signature analysis in patients with melanoma, and we observed that the patients with high enrichment of these cells, they have a better survival than the ones that have lower enrichment. And this is when you use the median. When you use the quartiles, you can see there is even a more dramatic effect. This is analysis univariate. So when you do multivariate analysis, you can also observe that TRM cells is the only factor that we analyze that can predict better survival or less probability to death in this patient, as in contrast with the stage that can go and produce a higher probability of death. So these studies have been shown by other groups that the TRM cells are quite important in different types of cancers. Like here I show you the ovarian cancer. As you can see, the TRM cell characterized by the expression of CD8 and CD103 are in tight contact with tumor isolates. In contrast with CD8 cells, there are CD8 T cells that are circulating that are not in contact with the tumor cells. If you look at the survival of this patient, as you can observe here, patients with no CD8 infiltration had this survival, and patients with the infiltration of CD8s that are not tissue resident also have a similar survival. But the patients that have an infiltration of CD8 T cells with this TRN signature marker, they have a better survival. So based on this data, we start studying the importance of these TRM cells in different human tumors. And we asked two different questions. So what is the phenotypic and functional heterogeneity of these TRM cells in tumors? And whether if these TRM cells are reactive against tumor cells. So for that, we started a project a few years ago in collaboration with Dr. Vincenzo Borna and a PhD student, Sophie Hidalgo, analyzing human renal cell carcinoma samples. So Dr. Borna was doing the surgeries of different patients, so providing us samples to analyze. And we could analyze the RCC tumors, but also in some cases, we also have the ability to obtain healthy tissue. When we analyze the cells that infiltrating these tissues, we observe that this is a flow cytometry analysis where you see the expression of CD69 and CD103 in memory CD8 T cells. You can observe that there is a clear population of double positive cells known as canonical TRM cells. But you can also observe CD69 positive cells that can also be part, a subset of TRM cells or perhaps activated T cells. And also you have the double negative cells that you can observe also in blood. So this is the quantification. As you can observe here, the most prominent population is this single CD39 positive cell. And TRM cells are quite frequent, as around 1/3 of the CD8 T cells that are infiltrating these tumors. And the double negative is a less frequent subpopulation. So when we analyze the phenotype of these cells, we observe this express, for instance, the canonical CD49A marker. Okay, so here analyzing by flow cytometry the surface expression of this molecule, and you can see the double negative have lower expression, and the single positive have quite high expression, but the double positive cell, the canonical TRM cell, have a higher expression of this, and this is the quantification, and where each point represents one patient. So this is a good marker to say that these cells are tissue resident. We also look for another marker like PD-1, which is associated with tumor reactivity. And we can also observe that TRM cells have higher expression of this marker as compared with the double negative cells. And we also did analysis of many other markers, but I will not show at this moment. But you can see that at least these two markers that are very important to define exhausted or dysfunctional T cells are also expressed in at least some degree in these two populations, as well as CD39. So another important markers that we are analyzing here is these two transcription factors. So TCF1, which is the TCF-left pathway associated with stemness, and the TOX1, which is the master regulator of exhaustion or dysfunction. So when you analyze these two markers, you can observe two different populations in the cells that are infiltrating tumors. So most of the cells are exhausted and are in a dysfunctional state, but you also have a few of them that are in a stem-like progenitor state, which is beneficial for the survival of the patients. So when you then analyze these two markers in the different subsets we are normally analyzing, we observe that the double negative cells have mainly these stem-like progenitors, while the single positive CD9 positive cells, they have mainly cells that are dysfunctional. In the case of tissue-resident memory T cells, you can observe that it's also both populations. So we believe at this moment that there are two states, or at least two subsets of these cells. One that is the dysfunctional state, and the other is the progenitor stemness that can give rise to this other terminally differentiated cell. So when you quantify this, as you can observe here, these cells have the highest proportion of these stem-like progenitors, while the other two have a higher proportion of the dysfunctional or exhausted CD8 T cells. So to better analyze these populations, we perform single-cell RNA sequencing in collaboration with Dr. Eliane Piaggio from Curie Institute. For this, we use the 10X platform, and we did the transcriptome analysis of single cells coupled to TCR sequencing, which can allow us to identify the relationship between these different subsets, and also to predict which of these cells are specific. So this is the differential gene expression analysis, which is a little bit difficult to follow in this presentation. So I will show you the UMAP analysis, where you can see the different clusters of cells that we could analyze. So we observe that at least a few of these clusters are TRM related. So we observe the classical exhausted TRM cells we found two clusters that have this type of gene signatures. But we also interestingly found these progenitor TRM cells that they are the ones that can express this stem-related transcription factor. And we also observed some TRM cells that are in active proliferation, and also circulating cells that proliferate and circulating cells that are progenitors. So based on this, we did clustering of these cells, and we at least observed that 3 or 4 of these clusters correspond to TRM cells. So in contrast with the initial belief, TRM cell populations are quite heterogeneous, and they do not represent a single cell subset. When we analyze the gene signatures previously published by other groups to analyze if they correspond to with our analysis in a similar way, we could observe that, for instance, this cytotoxic T cell signature has a really good correlation with our cytotoxic T effector memory populations observed. Other groups that have published signatures for TRM cells can also correlate with our exhausted TRM cells. And this is a proliferating TRM signature. We can also correspond to our proliferating TRM cluster. Interestingly, when we look for the TRM cell progenitors that we can we identify in this cluster number 7, this also correlate with a similar cluster observed by this publication. And the important thing is that this gene signature also correlate with positive response to immunotherapy. So we believe that the TRM progenitors may predict better response to immunotherapy. So to see if these TRM cells are tumor reactive, Sophia, our PhD student, set up a very nice assay. So we can obtain tumors and isolate the tumor-infiltrated lymphocytes and also the renal cell carcinoma cells. And we can culture these cells for a few weeks. But also, in the case of radical nephrectomies, we can also obtain the non-malignant renal tissue that we can culture and also for a few weeks. So then the TILs that we sort in a similar way where we analyze in these different 3 different populations, the double negative, the single positive, and the double positive TRM cells. And then after we have enough cells of each of these cultures, we can do co-cultures to see if these different T cells, CD8 T cells, can recognize the tumor cells. So for this, we did these co-culture assays, and we did either the TRM cell, the TRMs alone, or culture with the tumor cell. And as a control of the specificity, we use a blocking antibody that can block the HLA molecules presenting the different peptides. And also, we could use renal tissue as a control. So as you can see, observe here, we measure the activation of the CD8 T cell by upregulation of this 4-1BB marker. If you observe here, you see a nice upregulation of 4-1BB when the TRM cells are in co-culture with the tumors. And this is completely blocked when you use the HLA blocking antibody. Also, the tumors, the TRM cells are not able to recognize the renal tissue, indicating that this is a specific recognition between the TRM cells and the tumors. So this is the quantification of different patients. you can see here the TRM cells have a good recognition of the tumor cells, which is blocked by the anti-HLA1 antibody, and they do not recognize the healthy tissue. And the single positive cells also recognized, but in a lesser extent, and similar for the double negative cells. If you compare the three bars here that are recognizing the tumors, you can see here that the TRM cells are the ones that recognize with higher in higher frequencies the tumor cells. If we measure the production of cytokines in the supernatants, we can also see a specific production of gamma interferon and TNF-alpha, which indicate that these cells are not completely dysfunctional. So conclusions of this part is that TRM cells are relatively abundant in RCC tumors. The TRM cells represent a phenotypically and functional heterogeneous subset of tumor-infiltrating lymphocytes, and the TRM cells recognize autologous renal cell carcinoma cells, but not normal kidney cells. Okay, so our ongoing work is to address the specificity of the TCRs to see how these different clusters of cells are related among each other. So this is the second part is an ongoing project also where we are analyzing the TRM cells that are infiltrating the non-malignant renal tissue. Okay, so this is a juxtatumor cells, the juxtatumor tissue, and we observe that the TRM cells here are quite similar in phenotype since they express this canonical tissue residency marker, and also this marker that is associated with tumor reactivity. But they have a lower exhaustion and dysfunction, as you can observe here, the low expression of TIM3 and CD39. So this is quite interesting. So after this, we analyze the expression of the transcription factors, TCF1 and TOX1. This is just a reminder of what you can observe in the tumors. But interestingly, in the non-malignant tissue, there is no expression of TOX1, and all the cells express TCF1. So this is the quantification of different patients we have so far. But this is quite encouraging because this will indicate that the juxtatumor tissue is a reservoir of different TRM cells and other CD8 T cells that are quite progenitors and could give rise to the cells that infiltrate in tumors. So finally, what we did was to obtain these cells and to do the same co-cultures we did with the tumor-infiltrating lymphocytes. In this case, we also observed a quite good recognition of the tumors, which was highly specific because they did not recognize the non-malignant renal tissue, and they were completely blocked after HLA blocking. So this is the quantification of the 2 patients we have performed so far, and we observed this nice upregulation of 4-1BB. And also we observe production of gamma interferon and TNF-alpha. So as a conclusion of this part is that TRM cells in non-malignant renal tissue display a non-exhausted progenitor-like phenotype. That these CD8 T cells from non-malignant renal tissue recognize autologous renal cell carcinoma cells, but not normal kidney cells. Hence, we propose that the non-malignant renal tissue may represent a reservoir of tumor-reactive CD8 T cell progenitors that may positively impact clinical outcomes and have a therapeutic potential. With that, I just want to thank the people involved in this work, mainly Sofia Hidalgo and Vincenzo, which are leading this project with me. Diego Figueroa and Eduardo Roa, which are also involved in this project, and our collaborators, in particular, the team of Dr. Elian Piaggio, which is where we are doing the single cell RNA sequencing analysis. Thank you very much.
Thank you, Alvaro. I think it was very impressive and quite translational in the sense that you're working with the real tumors. So I think it's quite interesting. And also looking for non-tumor, which is the supposedly normal tissue, which is not real normal. So congratulations for that. Okay, I see that there's several questions for you to the end. So now we'll move to a final speaker of this session, who is Dr. Franz Villarroel Espíndola, who obtained his PhD in 2012 from Universidad Austral de Chile. Then he performed a postdoctoral fellow in enzymology and molecular metabolism with Dr. Juan Carlos Sleve at the same institution up to 2015. And then he moved to a second postdoc in translational immuno-oncology under the supervision of Dr. Kurt Schalper, who's a Chilean too, at Yale University in the United States in 2016. And at that institution, he was promoted as a social researcher in human pathology between 2017 and 2019. In April of that year, he decided to move back to Chile and took the position as a principal investigator and head of translational medicine laboratory at the Fundación Arturo López Pérez. His research line is the identification of biomarkers in solid tumors for the prediction of response to immunotherapy based on PD-1 and PD-L1 axis in the context of tumor microenvironment and the evaluation of cytotoxic immune response. Dr. Villarroel.
Hello, can you hear me?
Yes, now
okay, great. Thank you, Alejandro, for the invitation and for the nice introduction. I'm really glad with this opportunity to explain you and talk about a little bit how we are defining new biomarker for the analysis of solid tumors and how we can use that platform to develop new biomarker for immuno-oncology and immunotherapy. And I will use a little example using Vista protein just to make much easier the way that we are using to set specific biomarker, specifically in the lung cancer. So I'm having some issue with there. So currently, in the clinical aspect, we have several issues, mostly when we try to define the right way to assign a specific treatment. And so this is a very graphic way to see that we have the good, the bad, and the ugly in immuno-oncology and most in the cancer treatment. And maybe you know that in this moment, we have a very good therapies based on monoclonal antibodies that have improved a lot the benefits for patients that are in a very advanced stage of disease. And also in some cases with medbrain, they can survive and extend their overall survival for a long time. However, it's still, as a researcher, we don't know clear what is the real biology of the different biomarkers and how this biomarker can work as a predictor and define if those patients can really have a benefit after treatment. But the worst thing is that until now, the clinical endpoint that we are using for clinical trials, and also the way to calculate or estimate the balance between therapeutic and the side effect are really a mystery. And we need to know and put all together these three levels of information to define if we are giving or not a real benefits to our patient in a specific clinical trial or specifically in a hospital set. So I don't know if all of you know something about PD-L1 treatment as immunotherapy, but this is the most common example of molecule that have improved the benefits for patients. PD-L1 is an immunocheckpoint. It's a negative pathway that reduces and is working as an inhibitory molecule to keep the control of the T cells and reduce the extension of the lesions when you have a full inflammation. What is happening in the case of the tumor is that the tumor can overexpress this protein on the surface of the cells and then reduce the activity of the T cells and introducing this effect, this factor of the immune evasion. But what happened clinically is that using histochemistry, we can detect the level of this protein on the surface of the tumor cells. And using an objective view based on the expertise of the pathologists, we can define the specific percentage of positive cells and assign a specific treatment based on this level. So in that case, the abundance of this protein will define if the patient will receive or not in nivolumab or pembrolizumab or any other antibody that have been developed against this pathway. What is the problem is that the objective response still is not higher than the 40% in 35 different types of malignancies. And in fact, the presence of this protein is not only enough to classify patient as a responder, because there are people which they don't have PD-L1, also they have a rate response between the 3 and the 20%. What is the main problem is that we are forgetting that calculating in a very quickly way, if there is around 60% of people that even though they have the protein, they don't respond. And it is including actually now the information for nivo, pembro, durvalumab, atezolizumab, and avelumab. All of them are antibodies against this pathway. And in some way, obviously, we start thinking we are using one biomarker to classify patients. But also we need to analyze if we are using a really objective way to measure this biomarker. So what is happening is that here in this table, I'm showing you there are different types of clones that have been developed per companies or for some researchers to classify a specific cutoff for this biomarker. And they define the way to detect, the way to define positivity or negativity, and also classifying patients in different groups. All of them finally are going to the clinic and the industry to define the objective way, in some way, to classify our patients and receive a specific treatment based on the performance of those antibodies. So what's mean is that we are not being completely objective, even though the biomarker could be the best option. We are forgetting the balance that is happening in in the context of the full tissue. If we look at now what the different agencies are defining about what is a biomarker, we know that now any biomarker is a biomolecule able to be related with a normal or abnormal process. But when we look at now the other definition that we are using to set a specific procedure for medical assignment is that this biomolecule need to be measured and need to be measured in an objective way and need to be in some ways a procedure able to define an outcome or the disease or the incidence of that disease. What's happening now is that we know, and unfortunately the industry have produced a lot of different tools and machines to analyze in a quantitative way different biomarkers. So this is just an example of different platforms that are using fluorescence, like Agua platform that is a private service of the old university, a Virsa, that is a machine that we have already hit a FALP, Vectra Polaris, there's another machine using multispectral analysis with fluorescence, Umix from it's a new company that is developing a system using the kind of libraries and using barcode code to define the identification of the different biomarkers from nanostring. And then we have another type of machine that are using imaging mass cytometry or spectrometry and multi-ion beam analysis to create an image through the detection in this time of flight system. But when we start setting our multi-parametric platform for tissue profiling, we have now two different ways to go. We can use cells, FFP samples, animal models, and then we will have some limitation because we need to decide which platform will be our real element for analysis. So if we use fluorescence, we will have the problem of the mixing channels and also the number of biomarkers that could be detected simultaneously. Or if we go for the system using mass spec, we will increase a lot the resolution of our biomarkers, multiple molecules at the same time, but obviously it will increase the price and the cost for our laboratory. But we need to keep in mind that we are looking for these four aspects. We need to have a comprehensive view of what is happening. So in that case, the histochemistry is giving us the context of the tissue and the combination of the different type of cells in a specific moment of the clinical path of this patient, thinking probably that we are collecting samples from the diagnosis day until the different result after different treatment. So we can keep now and preserve the information based on the morphology of the cells and the tissue. It needs to be powered. It needs to be a powerful machine in the sense of to reduce the variability and increase the value of our sample, because sometimes we have a very little, very tiny piece of tissue and they cannot be used in serial section. Probably just we will have one option to do the analysis and finally improve the benefit of the patient. And the most important thing is that need to be reproducible. We need to have a very robust methodology to demonstrate that the biomarker that we are setting and proving will be able to apply it in the clinical stuff. Remember that we need to move from the research to the laboratory in a clinical environment to be over and over reliable for each patient that is being analyzed. Very generally, I will not explain you how works the protocol for the immunodetection in these platforms, but just I want to explain you that this actually is very easy. It's very common histochemistry, with the only one difference is that we are repeating over and over a procedure of staining, labeling, and bleaching or quenching, depending on which is the method that we are using. And in some ways, most of them are using microwave system, just reheating the microwave and removing one signal and going and going. But in our laboratory, actually, we are using a chemical system to detect different, using monoclonal antibodies against the different subclasses of IgG to detect all the combination in a simultaneous way for our different biomarkers. So we can combine from IgG1, IgG2, 3, 4, and so on in the same slide, in the same detection. But the key aspect, actually, in addition to the combination of platform that we can use, is that we need to be clear that we are trying to mix now two different worlds. The environment that the pathologist is using daily, in which he is using the expertise of the human eye to analyze a tissue section. But also we now to tell him and to the society and the medical environment that we can use a system and a supervised way to analyze the same concept. So all the machines in this moment, they have software and the ability to segregate the tissue in different compartments. So using cytokeratin, for example, we can create a tumor mask. That means all the tumor nests living outside all the area that is not tumor that we will call stroma. And then estimating the number of pixels that were positive for that mask, we can calculate the concentration in some way or give the identity and a specific pixel based on that information. So what I'm showing here is how we can analyze the real performance of our machine, comparing how was the result of the unsupervised analysis, in this case for the CD3, against how was the pathology in his normal procedure. Obviously, need to be the same technology, the same staining. If he is analyzing fluorescence, the machine need to be analyzing fluorescence. And we can see that it's a very high correlation between the expertise of the pathologist using the human eye and the machine. And also we can get this kind of a difference and level of quantification or number of cells count for both protocol, both procedures. But actually, the main problem in the translational research and then in the clinical aspect of validation and giving value to that data is the reproducibility. And this is a protocol that have been published years ago, and it's a very popular one. It's having a lot of downloads. And actually, this procedure is actually used in this moment for all the companies that are developing companion antibodies for diagnosis. This is used for cell signaling, for DACO, for Avcam, for Merck, to define the antibody that will finally reach the clinical laboratories. So what is it's very basic. It's a yes or no flow chart, and mostly is looking for sensitivity, specificity, and cross-reactivity. And then we can improve the value of this, introducing our own expertise as a basic researchers. But the most important thing at the end, it will be after the validation and after confirm that our machine is able to see the same as the pathologist, is that we need to introduce quality controls at technical gold standards. And in this case, the much easier way is having checkpoints and using arrays, index arrays with specific spot that can be compared day after day, being in different procedures of staining, and running this very basic correlation chart and see how my staining is working frequently, how is the performance of the technicians or the professionals that are doing the staining, and then defining the real value of this protocol. And the reason of having these three quality control and checkpoint is because at the end of the day, we want to say to the clinicians, this is the biomarker that you need to use to define if your people or your patient will respond or not on a specific treatment. And in that case, I will introduce now what is VISTA and how we took all that information that I was telling you before in the design and the development of this new biomarker for treatment. So VISTA is a new member of the B7 family. It's a kind of cousin of PD-1 and PD-L1, has a big homology in the extracellular domain, and is also a negative immunocheckpoint, and is related to inflammation, and in some way have shown anti-tumor response modulation through monoclonal antibodies. In some clinical models have improved overall survival of the different mice. And in this moment, there are some antibodies able to block VISTA in ongoing procedures to demonstrate if there is a real validity in the clinical assets. So what we did is obviously the first thing was the antibodies validation. And this is just the example of how we are taking the information that I show you as a theory, putting in the practical way. So the first thing is defining, obviously, that we can standardize the protocol. And the first thing is defining the optimal concentration of antibodies. So this is just it's a titration using our platform, defining the optimal amount of antibody to increase the ratio signal to noise, which it means, or increase the dynamic range to separate negative from positive cases. After having the optimal concentration, we need to define if actually we are using the right protocol. And in that case is we need to compare the antigen retrieval procedure to define if we can improve this dynamic range and carry on working still on the linear space of our dynamic curve. So in this case, just comparing high pH, low pH, in a specific concentration to define, okay, this is our best dynamic range. Knowing that, we go to sensitivity and specificity, and also we are using knockdown cells In other cases, we can use the peptide to pre-absorb the antibodies and define if we are not having cross reaction using naturally positive and naturally negative cell lines. Why we are not using tissue is because still the tissue has another background for the natural autofluorescence and also other technical aspects. So being clear that with a cell line, we have a very clear protocol, we can move finally to the clinical assays. And this is how we validate VISTA as a biomarker. In this case, we analyzed 700 cases of non-small cell lung cancer. All of them are part of a retrospective cohort collected in a specific period of time in the US and in Europe. We include stage 1 to stage 4 of this disease, including smoker, trying to keep the whole spectrum of patients that we can have in the US in that time. And also setting some aspect in the sense of the mutational status. And then obviously, because we wanted to see the performance of VISTA in the concept of different biomarkers together, we analyzed the co-expression of this protein in the sense of or the complexity of the PD-1, PD-L1 axis and the different subpopulation of T cells and the macrophages, and then the association of with the clinical outcome. So what we found is that this protein is a continuous variable, so we can quantify from 0 to something, and in both cohorts. So this validating now the performance of the antibody. So knowing that also it's a continuous variable, we can define a cutoff to explain and separate the population between positive and negative. But obviously, because this information is going to the pathologists, they need to have a visual pattern to be analyzed when they are running their normal procedure. So we define that most of the cases, more than the 80% of the cases, they have a very stromal pattern in which practically nothing of the protein is detected in the tumor's core, but most of them are in the stroma. And based on the morphology of the cells, we know that are mononuclear cells and mostly monocytes. But also there is a little portion, just only 20% of cases that have shown this pattern. It's a beautiful pattern in which we can see that some tumors are positive for VISTA, and showing this kind of a random detection of this protein. If we look at it more detailed, because the pathologists need to have the full range of staining possibilities, is that we confirm that actually most of the cases are stromal. There is some detection in the tumorness with different level of intensity and level of detection, but also there is a group of cases that they have a high level of infiltration, in which the tumorness is infiltrating dramatically by positive cells, but those cells are mostly immunocells, all of them practically monocytes. But we know that the tumor microenvironment is more complex than only tumor cells. So this is just a very, very classic representation how it looks a tumor. And then we compare the expression and the abundance of VISTA in the presence of different cells. So this is just the quantification of VISTA inside CD3, inside CD68, or inside CD20. And it means that clearly the T cells, they have higher level of VISTA, probably because it's also a smaller cell than the macrophages. And in the case of the specifically cytotoxics and Tregs, is the cytotoxic they have more, and probably that is explaining the value of this protein as a biomarker. But again, clinically, we need to say that is to the physician that is not only in a specific cell type, is also having a role in the biology of this patient. So we prove that high level of VISTA are related with a longer overall survival, and probably coming through this correlation with infiltration of T cells and also with the axis of PD-L1. Classically, yeah, thinking that VISTA could be a new biomarker and probably a new target for therapy, is we need to know which is the therapeutical window, or what type of people or patient will be really get benefit through this protein analysis. So using the basic uh, classification of people with the lung cancer is that we use the histology to define squamous cell carcinoma, or adenocarcinoma, and also the EGFR or KRAS mutation as a classifier. So what we did is this association with the clinical pathological properties, and all. The only thing that we saw is that VISTA is highly expressed in adenocarcinoma, mostly in the stroma version, and the squamous carcinoma is the only one that has the highest probability of having a mixed pattern. That means it's having VISTA in the tumor cell and the stroma. But really, this big difference is thinking in the mutational status of the cases, is that this is giving us the real light of which is the therapeutical window for this protein, because the highest proportion of cases with a mixed pattern of VISTA is in the mutated cases. And this mutation could be KRAS or EGFR. So just making a very quick conclusion about VISTA, it is highly expressed in lung cancer. It has a very unique immunostaining pattern that is related to the histotype and the mutational status. Is located in tumoral immunocells, so it's given probably as a very complicated view when we are trying to define the value of this molecule in the clinical concept of application. It correlates with a PD-1 axis that is giving us the probability to improve the benefits of antibodies against PD-L1, and is also is correlated with the longer overall survival. Our future perspective is obviously trying to move and push the system in the way to have more information. So one single biomarker probably is not enough. And in that case, we using another platform that is imaging mass cytometry, which is basically is using antibodies that have been conjugated with the rare metal of the nature, and using a laser that is ablating the tissue to create a cloud that is passing through a detector of time of flight mass spec. We can separate and combine more than 45 different biomarkers together. So what we did is we took baseline biopsies from patients that were in a protocol for PD-L1 blockade. We combined 29 biomarkers against different functional or phenotyping molecules and structural and biological processes to define which is the pattern of these cells. So then using a supervised system, we took the information of these 29 biomarkers together, making a new feature of analysis based on this information of people with the real benefits or without benefits after treatment. And we run an analysis and a supervised measurement of this feature in different type of cases using a training cohort and a validation cohort to separate people, and now with a completely unsupervised model in the people that will get benefits or will not get benefits after treatment. So it means that now that we can combine 20 markers and define which patient, starting from the first biopsy at the diagnosis, to define if that will be a real benefit in the future, if that person is going or not to an immunotherapy procedure. So in conclusion, just I would like to say is that there are several technologies that have been developed for discovery and validation of biomarker. The quantitative immunofluorescence, or the QIF, is just another tool which requires a reliable and robust procedure for an objective quantification. One single marker is not enough to describe cancer biology or predicting outcome. But if we can combine, it obviously will be more useful, but it will require more expensive technology and probably more analytical aspects to get a full result. And I would like just say thank you to all people that have been related to this research, starting for Kurt Schalper and David Rim in the US, and Miguel Samamer in Spain, and now the Translational Medicine Laboratory at FALP, which is where we are setting out all the same procedures for our patients. Thank you.
Thank you, Franz. Very interesting, very exciting too. Okay, I will ask all the speakers to turn on the video camera. We have 10 minutes for questions. So I'm going to start with a question to Dr. Fernandez. Since you identified a gene profile that characterizes several cancers and then two master regulator transcription factors that can now be targeted, any idea how this can be achieved without affecting normal function?
Can you repeat, because I can, I just allowed to open the microphone and the video. The video I cannot turn it on. I don't know why.
Okay, the question is, since you have identified a gene profile that characterizes several cancers and then two master regulator transcription factors that can be targeted, any idea how this can achieve without affecting normal function of these two master genes?
No, we didn't do this kind of experiments because we need to have normal samples, something that is usually lacking in this kind of databases. But it's a very good question. So I think it deserves to study these effects, but we need normal samples. Probably we can use the adjacent tumor samples available and take a look on that. It's a very good question. We will do that. Thanks for the advice.
Perfect. Ignacio Bigman is joined me for this roundtable. So next question will be for Ignacio. Go ahead.
Okay. So thank you, Alejandro, for your introduction. These were very nice talks by everyone. Congratulations to the 4 presenters. Due to time, we'll start by asking each presenter one question each, and then we'll go on with the other questions, okay? So the second question is from Paula Diaz to Vinicius, where she asks, is there evidence that RNA modifying enzymes change the regulation on non-coding RNAs between primary tumors and metastasis?
Hi, Ignacio. Hi, Paula. I did not find any evidence yet. This is a very new field. Only few groups are exploring the post-transcriptional modifications focused on non-coding RNAs. And I have not seen any work doing that by comparing metastases with primary tumors. This is something that we can do, actually.
Thank you. Next, the next question is from Charlotte Hill to Álvaro Yatza. Do you expect to see any differences on TCR specificity or on the dysfunctional TRMs?
I think this is a very good question. Thank you. It's known that when you have a high affinity TCRs, you have a faster way to become dysfunctional. So probably the dysfunctional TRM cells, they will have a skew repertoire for TCR to high affinity TCR. So we expect that perhaps there will be some difference, but we also expect that the same TCRs will be present in functional and non-dysfunctional TRM cells.
Perfect.
Great, perfect. The next question is from Alejandro to Franz. He asks, Franz, is VISTA expressed in other tumors beyond lung cancer based on the TCGA data and/or immune-associated tumors?
Yes, the answer is yes. Actually, VISTA have been published and detected in the renal cancer, gastric cancer, some type of breast is also naturally expressed in cells with the macrophage activity. So also some glia cells have expressed VISTA. So actually, it's a very well expressed cell, the protein, sorry, but the role is not clear. That is the main issue, because the intracellular signaling have been non-demonstrated yet.
Okay.
So, Alejandro, if you allow, I have one question for Elmer now, since there are no more questions directed to him.
Sure.
Elmer, how feasible would it be to use other gene signatures, mainly due to the onset of new single cell technologies that allow you to identify single cell signatures, right? How would you use those signatures for your deconvolution approaches, such as mixture, the one that you developed? That would be really interesting for immuno-oncology.
It's very simple. The problem there is not how to use it or of the possibility to use it. The main question is that we need to validate the signature and the algorithm in real data or in benchmark data. So if you have the signature and if you have some data that can be used as a truth, it's just very simple to test and to verify if the algorithm joined with the signature can be accurately used. So in this kind of problems, there is always a joint venture between the molecular signature and the algorithm. So we can test very easily if we have the molecular signature. We don't have them. We are not the group building molecular signature, but if the molecular signature exists, we can test it and we can build a model or the best model for this particular molecular signature.
Okay, so for example, Alvaro Latzer's signature may be the one that he used to identify those precursor CD8 cells. If that signature works, it could be used with a deconvolution algorithm on TCGA data, maybe.
Yes, that's right. See, if we have we need a gene expression molecular signature, and we can use mixture, or if they have a different molecular signature, a different type, we can build the model for these molecular signatures.
Okay.
Thank you. Okay, I have a question for Vinicius from another question. Do the changes in messenger RNA modifications in cancer affect RNA stability, or also the ability to be translated into proteins?
Hi, Andrew. Yes, not only that, but also affects splicing, localization, interactions with other molecules, let's say microRNA, mRNA, or microRNA, long non-coding RNA interactions. So this is a very huge new field and layer of complexity that it adds to our understanding of the transcriptional complexity of the cells.
Okay. So now I have another question for Alvaro. Actually, I was wondering, have you seen any differences between CD8 tumor-infiltrating lymphocytes between different TCGA subtypes? Because some of them have already some very interesting molecular signatures that can partly be correlated with the infiltrating or with the TILs. I've seen so in ovarian cancer, for example.
So your question is CD8 T cells from different types of tumors. They have different gene signatures, or?
No. My question is, have you used these deconvolution algorithms, these signature-based deconvolution algorithms on TCGA data to see if there are differences in CD8 cells between TCGA molecular subtypes? In, for example, ovarian cancer, I've looked for that, and I've seen that the group with the best response, which is the immunoreactive group, is also the one that has the highest CD8 infiltration.
Mm-hmm.
Have you observed any similar patterns on other types of TCGA data?
We're running those analyses at this point. For instance, for renal cell carcinoma, we took the classical TRM gene signature, and we observed actually opposite effect. So we see less, we see worse survival on the patients that have enrichment of the classical TRM gene signature. And this is because probably these cells are quite terminally differentiated. So now we are trying to curate our TRM progenitor signature and see if that can give us a better result. So it will be quite important to define what is the good TRM gene signature in our case, because as I told you, the same classical TRM gene signature when we analyze in melanoma patients, they have a really good survival. So there are some tumor-specific things that we need to analyze in depth.
Okay, there's another question to Vinicius. Is there any evidence that RNA-modified enzymes change the regulation of non-coding RNA between primary tumors and metastases?
Hi, Alejandro. I already answered this question. Ignacio did this question before.
Okay, so let's move to the next question. Ignacio?
Okay, this one's also for Alvaro Latter. Fabiola Sanchez asks, are there any differences among the binding of the different sets of T cells to the endothelium to infiltrate the tumor?
Well, it's difficult to answer that question because the tissue resident memory T cells are already inside the tumor, so they don't circulate, so they don't go to the circulation. So, but it's been known that the T cells after the priming, they are already committed to this subset. So it will be interesting to analyze if the different pre-committed precursors are already, they have already difference in the ability to bind to endothelial cells, for instance. But most of the cells that are effector-like, they already have the capacity to bind and to infiltrate. So, but it will be interesting to see if these pre-committed precursors have a differential ability.
Okay, thank you. It's already 10 a.m., so it's time to stop. So I would like to thank Elmer, Vinicius, Alvaro, and Franz for this system biology and immuno-oncology session of the ACDIS Spring Cancer Symposium. So I appreciate your talks, and I also appreciate the questions. I think it's very encouraging for these two already not novel, but in some way unique aspects of cancer research, emerging aspects, no doubt. Thanks a lot.
Thank you, everyone. It was a pleasure. Bye-bye.
Thank you.
Thank you, everyone. Thank you, Alvaro.
Thank you.
So I think now we'll have a break. Okay. Good morning, everybody. Can you hear me?
Yes.
So we will now continue with the keynote lecture of the day. Let me introduce our speaker, Marcelo Castañez. He got his PhD at the University of Buenos Aires in Argentina. Then went on to a PhD in pharmacology, by the way. Then 1990, 1995, went on to do a postdoctoral fellowship at NCI-NIH under the tuition of Peter Bloomberg. And there he worked on the regulation of protein kinase C, the interaction of C1 domains with lipids and activators. And that was actually a time at which the two of us were fierce competitors. Since then, we've become friends. He went on 1995 and joined the University of Pennsylvania as an assistant professor, moved through the ranks and became full professor in 2007. He is currently editor of multiple journals, such as Cancer Research, JBC, and also Scientific Reports and so on. Member of multiple review panels and advisory boards. And his main scientific research interests are in the area of cancer cell signaling, with a focus on protein kinases and small GTPases, particularly in solid tumors. And I believe that is what he will be talking to us about today. Marcelo?
Okay, can you hear me?
Yes.
Okay. So thank you very much, first of all, for the invitation, Andrew. As I said 3 years ago when I was in Chile, Andrew and I were fierce competitors, but he was always winning. I was always the loser. So he always published his papers first, and then I would come after that with my own papers. Anyway, but then it's true, we became good friends, and I'm very pleased to be part of this meeting and also see the names of a few friends and colleagues who are participating. So thanks very much again for the invitation. So I will share the screen. Oops, hold on. Give me a second. Oh. Can you see the screen?
No.
No, hold on then. Share screen. Oops, hold on. I'm having an issue here. It's not sharing, hold on. Oh.
Yeah, there we go.
Sorry, I'm really sorry. So, is that okay?
Yeah.
Yeah, I'm sorry. We use here another system, which is called Blue Jeans. And sometimes we have conflicts between the two of them. Anyway, so my talk is going to focus on cancer signaling by protein kinases and small GTPases. So relative to the talks that you had this morning, we are going to focus much more on cellular aspects and signaling in cancer, and how we can apply to identify eventually important proteins driving tumor formation, metastasis, and potentially acting as biomarkers for disease progression. So, hold on. Okay, so as everybody knows, cancer progression is a multi-step process whereby tumors are initiated, in a certain tissue, they grow, they expand, and eventually cells from the tumor may escape from the tumor, intravasate into circulation, then extravasate, and then form micrometastases that can eventually grow to form large metastases. And this is a very complex mechanism, as you know, that requires a very complex set of interactions between the cancer cell and the microenvironment, and also the ability of the cells to have a plastic phenotype by which cells can change the shape, acquire invasive capacity, and be able to colonize other tissues. And of course, we can view cancer from many angles, but the way we at least we are studying it is from a signaling perspective. So, well, cancer is a disease of dysregulated signaling pathway, which of course comes as a consequence of genetic and epigenetic modifications. And that involves changes during the transformation process in receptors, in ligands, in transducers and effectors, and in the pathways that ultimately lead to the transcription of genes and transcriptional programs that lead to transformation, and the plastic changes that I mentioned that eventually lead to invasion and metastasis. Of course, this involves the hyperactivation of numerous pathways or due to mutations, due to overexpressions, as well as the loss of tumor suppressor genes and tumor suppressive inputs that allow these oncogenic pathways to proceed. And in this particular talk, I'm going to focus on a family of kinases that was largely involved in cancer, which is the family of protein kinase C kinases. These are some of the most popular kinases ever studied, with actually more than 60,000 citations in cancer in PubMed. And these kinases represent a family of at least 10 or 11, depending on how you classify it, isoforms, which all have a kinase domains able to phosphorylate specific targets with a specific also pattern of recognition for the substrate, and a regulatory domain that dictates the classification of these kinases into classical or kinase or calcium dependent enzymes, the novel, which are calcium independent, and the atypical. And the main characteristic, at least of the two first families, the classical and the novel, they are the receptors for the lipid second messenger diacylglycerol, which probably you all know. It's in all cell biology textbooks. It generates through the activation of receptor tyrosine kinases or GPCRs, or through oncogenic inputs as well, and also through the activation by phospholesters. I remember by the time Andrew and I were young postdoctoral fellows, we were both studying how the phospholesters interact with the C1 domain doing the biochemistry. But then we really moved into the biology, and people started to think how these kinases relate to cancer, what pathways they activate. They're really related to many of the oncogenic pathways, as we will see later. So, and essentially these kinases became famous 30 or 40 years ago because, as I mentioned, these are the receptors for the phospholesters, and the phospholesters were widely used in a paradigm that was used in the 60s or 70s or even before, whereby people painted essentially the skin of the mice with a carcinogen, such as the MBA, and that leads to the first stage, which is the initiation. And then when you apply in a repetitive manner with a certain frequency the phospholesters, then the mice started to develop papillomas, which eventually they may convert to malignant carcinomas. So that made the link of PKCs with tumor promotion a very well studied phenomenon through the years. What I'm going to focus this talk now is, or today, is in the fact that PKCs have role beyond the promotion stage. We believe that these kinases are really important in certain circumstances and certain cancers in the initiation. And also, I'm going to show some evidence for the role of these kinases in the late stages of cancer, particularly in invasiveness and in metastasis. And this is really important because it is possible to target these kinases with inhibitors, although people have been studied inhibitors for quite a while. The initial studies were not very successful in cancer, particularly because this is a large family of kinases that they have, in some cases, opposite roles. Some of these kinases could be tumor promoters, and some of these kinases indeed turn out to be tumor suppressors. And there is a significant literature and a number of papers recently in the last years in Cell and other prominent journals showing tumor suppressor genes for PKCs. So this is complex from the pharmacological standpoint because people have not been really successful in generating at least ATP-based inhibitors that can discriminate between these kinases. But I think that there are a lot of opportunities to do that. So it is very clear that PKCs, unlike other pro-oncogenic kinases, are generally not mutated unless in certain types of tumors, such as some rare tumors and some subclasses of gliomas, in which mutations in PKCs can actually drive the disease. But in most cases, what really contributes to cancer progression is the activation status, which is not very easy to measure compared to other signaling molecules, and also the actual expression levels. So there is a significant change in the expression of some of these kinases and a shift in the balance between tumor suppressing and tumor promoting PKCs. One of the best examples of the oncogenic PKCs is PKC epsilon, which is overexpressed in a number of epithelial cancers, such as in prostate cancer, in lung cancer, in head and neck cancer, et cetera, et cetera. This is just some example that we've done in collaboration with the Department of Pathology at the University of Pennsylvania years ago, where we measure the expression of PKC epsilon by immunofluorescence, by immunohistochemistry, I'm sorry, in cancer versus adjacent tumors. And you can see that there is a significant overexpression of PKC epsilon that actually correlates with expression of active NF-kappa B and also COX-2 and several other markers related to progression of cancer. The same can be recapitulated in cell lines. For example, these are very aggressive prostate cancer cell lines that they have very high levels of PKC epsilon. They correlate with COX-2, and they correlate with activation of NF-kappa B, so nuclear NF-kappa B, and loss of cytosolic NF-kappa B. So one of the questions is whether the overexpression of PKC epsilon, as can be seen in cancer, can actually recapitulate or can induce or trigger the development of cancer. And years ago, in order to address this question, we generated a number of transgenic mouse models where we overexpressed individual members of the PKC family in the prostate under the control of a probasin promoter. It's an androgen responsive promoter. And as you can tell, only PKC epsilon was found to cause a severe phenotype in the mice. Actually, when the mice become sexually mature, and this is an androgen responsive promoter, they start to develop hyperplastic phenotype, and eventually that leads to low-grade and high-grade PIN, which are preneoplastic lesions. But in any case, we were able to find actual cancer, so we were really not able to find it. So, but we have actually 100% incidence in the number of hyperplasia and PINs in the PKC epsilon transgenic mice. However, what we found later is that PKC epsilon by itself, even if it's not able to be an oncogenic driver by itself. It really cooperates with other oncogenic inputs, for example, with loss of the PTEN tumor suppressor gene. So when we cross the PKC epsilon transgenic mouse with a mouse which was haplodeficient for the PTEN tumor suppressor gene, so losing only one allele of the gene, which by the way, by itself gives a very similar phenotype as the PKC epsilon transgenic mice, the combination of both overexpression of PKC epsilon and loss of the PTEN tumor suppressor gene leads to the development of a very aggressive prostate adenocarcinoma with more than 50% incidence. And actually in those adenocarcinomas, we could observe, as you could see here, significant nuclear NF-kappa B, activation of the NF-kappa B pathway, high COX-2, and also several other markers such as very high levels of phospho-AKT, S6, mTOR, phospho-ERK, STAT3, and phosphostat3. So clearly, PKC epsilon could have a role in the initiation of the disease and also in the progression and eventually contributing to the development of highly invasive adenocarcinomas. We went on to work on the mechanisms driving this phenotype, and I don't really have the time to show it, but essentially we identified two pathways that were key effectors. One actually involves COX-2, so PKC epsilon in conjunction with PTEN loss promotes the activation of COX-2 and generation of prostaglandin E2, which leads to probably an we believe it's an autocrine loop through the PGE2 receptors in the prostate cancer cells, and that contributes to the tumorigenesis. And it's very interesting that if you feed the mice with rofecoxib, a COX-2 inhibitor, for several months, when you sacrifice the mice after a year, this is a very slow progression model, you can actually inhibit completely the formation of carcinomas, even the invasive adenocarcinoma. There is also a significant reduction in CD31 staining, which is angiogenesis. And if you actually measure angiogenesis by measuring the mean vessel area, you can see a significant inhibition by the COX-2 inhibitor. So essentially we identify two pathways which are represented here that involves the NF-kappa B non-canonical pathway and the canonical pathway. The COX-2 pathway leading to PGE2 production and acting through the EP receptor actually works through the canonical NF-kappa B and also through bioinformatics and a lot of functional studies. We published a couple of years ago a paper showing that the PTEN loss, which leads to PI3 kinase activation in conjunction with PKC epsilon overexpression, leads to the activation of a non-canonical NF-kappa B pathway, which ultimately impact on the promoter of CXCL13, which is a chemokine that is produced by cancer cells. And actually that acting through the CXCR5 GPCR receptor leads to an autocrine loop driving both growth, migration, and invasiveness. So changing or switching to another type of cancer, we also became very interested in non-small cell lung carcinoma, because actually it has been reported years ago that there is a very high level of PKC epsilon over expression, really an aberrant hyper expression in the tumors of non-small cell lung cancer cells, not in the small cell, but yes, in the non-small. Actually, when we started to look through bioinformatics, we couldn't find really significant differences in overall survival of the patients when we categorize the patients into low or high PKC epsilon. But when we started to discriminate based on a specific driver mutations, we started to find very interesting results, particularly the fact that those patients that have mutant KRAS, they have a much worse outcome than those patients that when they have very high levels of PKC epsilon compared to those patients that have low levels of PKC epsilon, suggesting a functional link between mutant KRAS and PKC epsilon potentially in driving the disease. So the idea was, can we recapitulate that in an animal model? So in order to do that, we generated a mouse model in which we express mutant KRAS in the lungs of mice and cross it with a PKC epsilon knockout mice. And then we can develop the KRAS tumors by intratracheal inoculation of adenoviral Cre that promotes the expression of the KRAS, the mutant KRAS allele. And then we analyze lesions 32 weeks later, and also we've done a 2-year survival curve of these mice. And very interestingly, the mouse that have normal high PKC epsilon, they develop tumors significantly. However, losing 1 or even 2 alleles, or better with 2 alleles, essentially there was a major reduction in the formation of lesions in the lungs. And if you translate that to a Kaplan-Meier survival curve, you can see that eventually we could recapitulate what we see in the patients. Those patients with, as I mentioned before, with mutant KRAS and high PKC epsilon, they have a poor survival, as you would expect, compared to those lacking PKC epsilon. We're really working now on the mechanisms to understand why this is happening. In a second model, which is very well-established model, we use a carcinogen, benzopyrene, which, as you probably know, induces the development of lung tumors. And so what we've done is first we have to change the background of the mice to AJ, which makes it sensitive to carcinogens. It's a 2-year process almost. And then we treat it with benzopyrene, and then we analyze the formation of lesions. And as you could see here, it's very interesting to find that these are the mice which have the 2 PKC epsilon alleles. They form tumors regularly. You can see here the total number of lesions, hyperplasias and adenomas, and however, losing 1 or 2 alleles in particular leads to a significant reduction in the formation of lesions. So, really arguing that PKC epsilon was really required for the development and probably the initiation of tumor formation in lung. So switching now into the late stages. So we believe also that this family of kinases play really important roles in late stages of disease. In particularly, we found years ago, and also several other people reporting in the literature, the involvement of PKCs in the migration or cell motility of cancer cells. So a simple experiment in which you knock down PKC epsilon using RNAi, you can see that leads to a significant reduction in migration, either by Boyden chamber or by classical wound assay. And when we started to look at the mechanisms, we found that actually PKC epsilon is really important for the formation of membrane ruffles and lamellipodia, which are required for cancer cell motility. So, for example, in this particular experiment, we treat the cells. We managed to develop in collaboration with medicinal chemists an agonist, DAG-like compound that has a prominent activity towards PKC epsilon relative to other PKCs. And as you can tell, this induces the formation of ruffles. This is a typical 3D picture of the ruffle, which interestingly, it's not a dorsal ruffle, it's more like a peripheral ruffle and also a ventral ruffle. We believe that this could be related to the formation of invadopodia, and we have some evidence for that. Essentially, when you knock down PKC epsilon, you completely lose the ability of the cells to form ruffles in response to PKC epsilon activation. So when we start to work on the mechanism, then we had to go, the obvious question, at least for us, was that we had to go to the effectors that drive the formation of lamellipodia and ruffle. And as many of you know, I know Andrew was working also on small GTPases. One of the key small GTPases driving the formation of motile and invasive lamellipodia and ruffles is the small GTPase in RAC, which is a which cycles between a GTP inactive and a GTP active form. And the activation of RAC is driven by a family of very large proteins called GEFs, or guanine nucleotide exchange factors. So we believe that the link between PKC when it's activated by phospholesters or by growth factors is through GEFs. But first we have to really check that this is RAC dependent. So we generated using CRISPR non-small cell lung cancer cells lacking RAC1, and as you can tell, the formation of ruffles by growth factors or by the phospholester essentially are lost when you knock down RAC, when you knock out RAC, using CRISPR. And interestingly, also we found using a Fred biosensor that RAC activation occurs in the ruffle. So when you stimulate with a growth factor or with PMA, you see a significant activation of RAC in the peripheral ruffles. So that really indicates that PKC activation has to be linked to RAC somehow. However, the identifying the effectors driving motility and eventually metastasis, and I forgot to mention that knocking PKC epsilon also causes impaired metastasis in using lung colonization assays in mice. Identifying the RAC-GEFs is extremely complex because there are at least more than 40 RAC-GEFs described, and they're really large proteins with a multitude of roles and really very complex biology. So how do we tackle this issue, this problem? So we generated in the lab what is called a RAC-GEF array, where we can profile in one qPCR analysis all these 43 GEFs in just one shot, and we've done that in multiple lung cancer cell lines with different mutations, KRAS, EGFR, ALK, etc., and we can actually get a profile. We can get the delta CT. So these are the very high expressing GEFs, and these are the very low expressing GEF. And then once we profile it in any given cell line, we can do it in lung cancer, and we've done it in many other cancers as well. So we can start knocking down one by one. It's very tedious, but but in the end, we managed to get results. So we can knock down at least the top candidates, and then we can measure ruffle formation in response to growth factors. And this gave a very clear result. We identified only 3 RAC-GEFs in non-small cell lung cancer cells, which are FARB1, RGF39, and TM2, as responsible for ruffle formation in NSCLC cells. And we have to say that one of the concerns with this type of assays is that, of course, that there could be compensatory effects between different GEFs. When you knock down one, you can get compensation of the other, and you won't be able, of course, to knock down 16 or 18 all at once. But these results clearly show that at least for these 3 GEFs, there is no compensation whatsoever when you do a single knockdown or knockout by CRISPR of these particular GEFs. When we measure actually migration and invasion, we found that the 3 of them actually were individually required for motility and invasion. So when you knock down RGF39, FARB1, or TM1, you can see a significant reduction in motility. This is a migration assay in living cells. Or if you do an invasion assay in using a Boyden chamber with matricel, then you find significant inhibition of invasion when you knock down any of these RAG-GEFs. I don't have really time to go through the mechanistic part. We're working on the signaling pathways, and actually we're actually close to submission of this manuscript. But what is really interesting when you go to the bioinformatics and doing this is in an unbiased way, you found that of the 43 GEFs, only 6 RAG-GEFs are massively overexpressed in some cases in tumors, in subsets of tumors compared to normal. And that includes FARB1, RGF39, and TM2. And actually, this is a partial picture, but we initiated a collaboration with the Department of Surgery. We're getting fresh tumors, and we can isolate EPCAM positive cells from the primary tumors and measure by qPCR expression. And you can see significant overexpression of RGF39 and FARB1. It turned out that these two actually overexpress much more when we have a larger number of samples. So we believe that there is a link between oncogenic inputs, or could be RAS, or could be growth factor receptors, via diacylglycerol. This is PLC gamma 1, the enzyme that generates DAG, activation of PKC epsilon, activation of RAG-GEFs, activation of RAG, and eventually that leads to actin cytoskeleton organization, formation of ruffles, motile structures, and invasion and metastasis. We are currently generating mouse models for these RAG-GEFs to see if we can recapitulate what we see with PKC epsilon. So in the last 5 minutes or so, I want to show also other interesting twists that we have with with PKCs. In this particular case, in triple negative breast cancer, in collaboration with a laboratory in Cordoba, in Argentina, we were working on the interactions between PKCs and EMT, epithelial to mesenchymal transition, a plastic program that converts the epithelial cells into highly invasive and motile cells. And we observe a significant correlation between the expression of PKC alpha in this particular case and SEB1, which is an EMT transcription factor. If you just go to cell lines, what you see actually that the cells that express high SEB are those of basal-like or triple negative, and there is significant correlation between expression of SEB1, expression of PKC alpha, and also those cells that have the mesenchymal phenotype, high vimentin and low E-cadherin. And actually, if you knock down PKC alpha from triple negative breast cancer cells, what you see is a significant reduction in the expression of SEB1. So PKC alpha controls SEB1. We believe that involves both transcriptional and non-transcriptional mechanisms. And we reported that last year, part of that in a paper. And actually, if you start doing more correlation analysis using bioinformatics in cell lines and in patients, you see that in this particular case, in triple negative breast cancer, there is no correlation between PKC epsilon and PKC delta. But when you go to PKC alpha, the yellow dots, the yellow circles are triple negative. They have much higher levels of PKC alpha, much higher levels of SEB1. And interestingly, they have also very high levels of AXL, which you probably know is a tyrosine kinase receptor, very important in EMT, very important in immune evasion and in drug resistance. And also we found a significant correlation between PKC alpha levels and CD274, which is the gene for PD-L1. So the immune checkpoint ligand. So we started to investigate where there is any causative correlation or association between these players. And also, when you actually go to the cell lines, you see that the cells that express the basal-like, that express higher levels of PKC alpha, interestingly, they have very high levels of phosphoaxel, also very high levels of total axel, so more axel and more activated axel. And also they tend to have higher levels of phosphorylated PLC gamma, which is the enzyme that generates DAG. So probably there is high production of the lipid second messenger that induces PKC alpha activation and then eventually a phenotype. It's very interesting that if you analyze the signatures of transcriptional signatures between PKC alpha and AXL in basal-like breast cancer using databases, what you find is that there is a significant overlap in genes from patients that have high PKC alpha and high AXL, or down and down. And in particular, there is a significant number of cytokines and chemokines, modifiers of the tumor microenvironment that are overlapped in this signature. And when you go to analyze the geobiological processes and the pathways, you see actually significant, in the shared signature, a significant number of programs related to ECM, integrins, cytokine, invasion, MMP activity, and pathways that also relate to these processes. So I think this is a very, this is preliminary, but I think it's driving our couple of next years of our research. And interestingly, those cells that express very high PKC alpha, they also express very high levels of PD-L1. And this suggests the possibility that elevated or aberrant activation of the DAG PKC alpha pathway leads to immune evasion, or is a tumor cell intrinsic mechanism driving immune evasion. Although we do not have yet formal proof for that, I really want to show it, because when you knock down PKC alpha from triple negative breast cancer cells, you see a significant reduction in PD-L1 level, both at the protein level, at the mRNA level, and I'm not showing it here, also using a promoter, a reporter for the promoter of PD-L1, CD274, we see significant reduction when you knock down PKC alpha. And also in this paradigm, we found that when we knock down AXL, also we see a significant reduction of PD-L1, which has been also described in other type of cancers. So the paradigm that we are investigating now and that we are really very excited is that in triple negative cancer cells, there is hyperactivation of PLC gamma 1, which may occur both through activation of receptor tyrosine kinases, by heterodimerization between AXL and receptor tyrosine kinases, or probably it's not very clear whether AXL itself can signal through PLC gamma 1. The literature is very ambiguous. But that actually generates DAG, activates PKC alpha, and we found that PKC alpha controls probably at the transcriptional level the expression of AXL, the expression of PD-L1, and there is probably a very complex set of interactions that we are working these days, and ultimately leading to the production of chemokines and cytokines and a high expression of PD-L1. So this shows very clear that PKCs have a role in metastasis. They could be contributing to lamellipodia formation, to invadopodia formation. I'm not showing it here. They could contribute to EMT, to protease production. We found also production of metalloproteases are regulated by PKCs, also anoikis, which is very important in cancer, and eventually drug resistance and immune evasion. So, so this is I think the last slide. It is very clear to us that PKCs have crucial role beyond the promotion state. So in promotion, it's very clear that the activation of growth-promoting PKCs or inhibition of growth-suppressing PKCs contributes to the promotion. Initiation in some tumors, loss of function of PKCs or gain of function of PKC mutations can contribute to initiation. There are also very rare cases of gene fusions, but also the upregulation that occurs actually the expression of certain kinases such as PKC epsilon is required for the development of the tumors. And also it became very clear that PKCs have a role by cooperating with oncogene or loss of tumor suppressor genes to develop invasive phenotypes, and also for the activation of pro-invasive phenotypes. So with this, I want to thank you for your attention, and these are the main people that contributed to the work that I showed today. Thank you very much.
Okay, thank you, Marcelo, for the presentation. Very interesting indeed. We have a number of questions in the chat here. So, well, I'll start off by asking you a more general sort of question. In your overexpression experiments using the different isoforms, let's say PKC epsilon, I mean, you were just using a wild type protein, right?
Mm-hmm. Yes.
So do you really know that the activity of the protein is required? Did you ever do an experiment using, for instance, a kinase inactive PKC epsilon or something like that? Or is, for instance, regulation by lipids required? Did you look at a mutant in the regulatory domain, in the C1 domain, and so on? Well,
so we use the wild type. It's been essentially impossible, even using inducible systems, to express a hyperactive PKC epsilon. The cells do not tolerate it. It's unfortunately, we do not know, as you probably know, it's very hard to know or to measure hyperactivation of PKCs. This is not like when you measure ERK or AKT with a phosphoantibody. There is the phosphorylation of PKCs do not necessarily correlate with the activation status, other than a very few exceptions. So it's hard to know whether PKC is activated. We believe it has to be activated in certain cases, because we do see, at least for breast cancer, the hyperactivation of PLC, which is the enzyme that generates DAG. That should translate into that. And also, in some cases, we do see enhanced membrane expression of PKCs. So which, as you know, translocation to the membrane correlates with activation. So we believe that there has to be, in certain cases, hyperactivation of PKCs. Not just, I mean, just the overexpression is, you need an enhanced input. Many oncogenic inputs, such as even very old papers, as you probably know, in the 80s, show that mutant KRAS leads to enhanced generation of DAG. So you have an oncogenic, an input via the lipid signaling that leads to PKC activation, or there should be, it should be there, I believe. Okay, let me move on to some of the questions in the chat. So it's a question here by Fabiola Sanchez, and she's wondering whether nitric oxide regulates PKC epsilon. And also she's wondering whether you've done anything looking at the role of PKC zeta in cancer. The first part, I don't really know. I don't remember seeing any type of modification. So PKCs are regulated through oxidative processes. There is some old literature. It's not clear at the physiological level or in cancer if this really happens. And with NO, I don't really know. PKC zeta, it's another world. It's not the acylglycerol regulated. It's regulated through protein-protein interactions, P62, and via phosphorylation mechanisms, and leads to a completely set of different. So it's part of the PKC family, but it's belongs to another class. So there are multiple a paper showing that PKC zeta is actually a tumor suppressor gene in colon, for example, the studies from Jorge Moscat, and also IOTA, which is similar to zeta, papers from Alan Fields, many other people show that actually it's an oncogenic. It's expressed in, it's overexpressed in multiple cancers, in ovarian cancers, and drives in lung cancer as well, and it drives tumorigenesis.
Okay, thanks, Marcelo. Then we have a question here by Lisette Layton, wondering whether the effect of rofecoxib in prostate cancer would also work in a normal PTEN background. So basically, is there a connection there between the?
We reported that in the so this is published in 2018. So the COX-2 inhibitors, they have a prominent effect only when you get the COX-2 overexpression. If you do not have PKC epsilon overexpression and you have wild type PTEN, COX-2 inhibitors have no effect whatsoever, which makes sense because there is no COX-2 overexpression. There is no effect at all. Yeah.
Okay, thanks. Then the next question here, Jorge Diaz. How do you think PKC activates RAC-GEFs in your model? Are there post-transcriptional modifications, or do, for instance, the PKC simply promote recruitment somehow to membrane ruffles?
Well, we believe it does promote the recruitment. We don't know whether this is a direct phosphorylation. There are studies with certain GEFs showing that phosphorylation is crucial for relieving the autoinhibition of RAC-GEFs. Most RAC-GEFs are in a close conformation, for example, the VAV1, and tyrosine phosphorylation in that case causes a switch and the activation of. We don't know. Two of the RAC-GEFs that we identified, they have putative PKC phosphorylation sites. So that's the next obvious question. These are very fresh results. We haven't gone there yet, but we believe it's either direct phosphorylation or maybe involves some other proteins in a complex. That's another possibility that we are investigating, or involves some PKC downstream kinases, because PKCs affect other kinases that may ultimately impact on the RAC-GEF. We don't know yet. We are doing now, well, the next step is to do phosphoproteomics and analysis of phosphorylation sites in the GEF that we identified. We are doing that now with FARB1. And once we have the result, we'll get a better feeling whether it gets phosphorylated or not in response to growth factor and PKC activation. Yeah, but that's a very good question. Yeah.
Okay, Marcelo, there's another question here by Marcela Hermoso, and she's wondering whether the activation of CD pathway or PKC dependent signaling, whether this leads to transcriptional regulation of PDL1 CD274 via NF-kappa B activation. Is that clear? And the other thing is, do you see production of any specific cytokine profile related to tumor immune activation?
I'm not sure I have the slide here. No, I don't have the slide here, but in this talk. But yes, there is. If you do, we've done RNA-seq for cells in which we manipulated PKC alpha expression, and we see a profound change in chemokines. I had that slide. Unfortunately, I didn't include it in this talk, and those results also correlate with changes in certain chemokines and cytokines. That includes interleukin-6. That includes well, some other chemokines. I have a list, both in human samples that correlate with very high levels of PKC alpha. So there is correlation in the signatures as well, and in the RNA-seq that we developed. So the answer is yes. Whether NF-kappa B is involved or not, we don't know yet. It's a logical target. It's not that NF-kappa B is implicated in tumor intrinsic expression of PD-L1 in some models, but we don't know yet. But this is the logical direction as well. Yeah, thanks for the question.
Okay. Then we have another question here about whether PKCs are in any way related to, since they promote migration basically, are they in any way related to extravasation of tumor cells?
Probably yes. Not sure in which we reported in prostate cancer that PKC epsilon is really important for seeding in bone metastasis. We use a model of bone metastasis that we published about 4 or 5 years ago. And when you knock down PKC epsilon in that particular case, very aggressive prostate cancer cells are unable to extravasate into the bone, and we can actually count and flush cells from the bone marrow and found that there are less cells when you knock down PKC epsilon. So we do believe that is happening. I don't know the mechanisms though.
Mm-hmm. So there's a question here relating to the role of PKC epsilon in cell migration. Is its activation somehow connected to integrins? If so, which one?
I don't know. We do see changes in integrins in all this profiling that we do, but we haven't done any functional study. Yeah. So I don't know.
Okay, and I'm being told here that we have to finish the session here. So just one last question here in terms of whether you think that PKC could potentially be targeted for therapy using, let's say, like nanotechnological applications. Are you doing anything in that respect?
Yes, I do believe that some of these PKCs may be targeted. So far, the major issues were with the development of really isozyme-specific inhibitors. But there's a study, for example, with a compound developed by Pfizer that targets PKC epsilon almost specifically, and it has very strong effects in KRAS mutant colon cancer. So yeah, I think it is possible. And it requires a lot of medicinal chemistry. But you know, the ATP binding site is very similar among the PKCs, and that's a major complication that we have in the field. We have to probably target other domains.
Okay, Marcelo, thank you very much for your presentation. Very interesting indeed. Stimulated a lot of questions. Unfortunately, we have to move on to the next. Yeah, thanks.
Thank you.
Okay, so we're going to move on to the next session now. With Marcelo Kogan. And so I will introduce Marcelo. So he's going to be chairing this session, and he's also going to be the first speaker in this session, which will deal with different sort of nanotechnological applications and how they might be useful in cancer. Marcelo Kogan is also Argentinian. He's a biochemist and pharmacist, got his degrees from the University of Buenos Aires, and then got a PhD in organic chemistry from the same university. He then went on to do his postdoctoral research at the Institute of Biomedical Research, Barcelona, and worked there in on the administration and use of peptides and different types of applications. His interest is essentially now centered on this type of application using nanobiomaterials. I think that is what he's going to be talking about today, specifically in the area of Alzheimer cancer and also cardiovascular disease. So Marcelo Kogan,
thank
you. Go ahead with your talk.
Thank you. Okay, thank you, Andrew. Yes, as Andrew mentioned, we will start the last section of the meeting. I encourage you to make highlights because at the end of the section, we will talk about highlights of each section. In this section, we have 4 talks. The first one is my talk. The second one will be a talk by Nelson Duran. After that, Nelson Duran will give the talk that was supposed to be given by Wagner Favaro. He has a problem and he could not give the talk. Nelson works in collaboration with him and he will give the talk. And finally, we will have the talk of Dr. Silke Kroll. I will start with my talk. Okay. Yes, my talk is nanoparticles for theranostics of cancer. Important issues from the point of view of pharmaceutics are relevant for diagnosis and therapy, as for example, stability of drugs, the formulation for in vivo administration, solubility of drugs, the drug delivery, control release, the selectivity, the targeting, as we mentioned in the last talk by Marcelo, the toxicity of drugs, the side effects, and other parameters. All of these are relevant to be considered for therapy and diagnostic of cancer and other diseases. In our case, we are using nanotechnology to contribute to the therapy and diagnosis. We are using different kinds of nanoparticles, polymer nanoparticles, dendrimers, gold nanoparticles. We have in the Advanced Center of Chronic Disease a group of different researchers that can cover the different kind of nanoparticles for applications. Other concept important is the concept of theranostic, in which the same nanomaterial or material could be used for detection of the tumor, to image the tumor, to detect, to do the diagnosis of the tumor, and to treat the tumor, by, for example, by irradiation at the specific place. This is a contribution of nanomedicine that in which we are working. Why nanoparticles can improve formulation of drugs? Nanoparticles could improve solubility and stability of drugs, as can be seen here. This is a project carried out by Felipe Garzón in collaboration with myself and Andrew Quest, in which Felipe include in a nanoemulsion curcumin. The idea of this curcumin is a very active drug that is hydrophobic and unstable, and he included the curcumin in this nanoemulsion, and we can see that the inclusion in the nanoemulsion improve the stability of the drug. We can see here in green the concentration of the curcumin included in the nanoemulsion after irradiation with UV light. This is an example, a nice example of how nanoparticles can protect a drug and improve solubility. Other point is important, the size of the nanoparticle. The interaction of nanoparticles with cells allow to produce internalization in the cells and cross barriers as gastrointestinal barrier and or other barriers as the blood-brain barrier. Nanoparticles can enter to the cell by endocytosis and can cross the cell and the barrier. Also, we have here in this picture another nanoparticle, natural nanoparticle in which we are working now, are the exosomes. Exosomes are nanovesicles produced by the cells that play an important role in the communication between cells. The interesting thing is that exosomes could have a kind of tropism to different kind of cells or places in the body. In this sense, we can use exosomes to improve drug delivery of drugs, as we will see next. Other important point is the passive targeting due to the abnormal irrigation of tumors. As we can see here in the tumor, we have abnormal irrigation, and nanoparticles could be penetrate to and be concentrated in the tumor, which allow us to produce a passive targeting. We can include the drug in the nanoparticle, and in this way, the nanoparticle can accumulate in the tumor, and we can concentrate the drug in the tumor due to the passive targeting that we can see here. We can also functionalize or cap the nanoparticle with a molecule. For example, we use folic acid. Some cancer cells overexpress the receptor of folic acid. In this way, we can address the nanoparticle to specific cells. Other interesting nanoparticles are magnetic nanoparticles, in which we can concentrate the nanoparticle with a magnet in a specific place in the body. Finally, we can talk about the controlled release that we can produce with nanoparticles, therapy and theranostics. We work with gold nanoparticles. The idea is that after we can irradiate the nanoparticles can absorb the energy and dissipate it as heat. The heat can destroy tumor cells, and in this way we can produce the localized therapy in a specific point. On the other hand, the heat can allow us also to produce the release of drugs, as we will see. On the other hand, there is interaction between light and nanoparticles, and we can have also other effects as scattering. In this way, scattering could be used for detection of nanoparticles. In this way, we can use the nanoparticle for theranostics, therapy and diagnosis. I will give some examples that we develop in ACDIS that improve formulations and allow also the theranostics of tumors. In the first case, we will talk about gold nanoparticles for spatial and temporal release, control release. We have an example here in which we use methotrexate. We obtain an inclusion compound with cyclodextrin, that is also a molecule that is used to improve drug delivery. And also, we include here gold nanoparticles. We obtain this ternary complex. After irradiation, it's possible to disassemble this complex and produce the local anti-tumor effect in HeLa cells. The problem that exceeds most of the formulations is that present a specific release of the drug. This is due that the drug is not covalently bind to the nanoparticle. In this case, we develop a methodology in which we bind covalently a molecule as an anti-tumor drug to the gold nanoparticle, and here we have a thermoclivable molecule that in the presence of heat can be cleaved and release the drug. We can do that with irradiation. In this way, we can produce the control release in a specific place in the body. This work is doing in collaboration with Eileen Araya from the Universidad Andrés Bello. We have other example. This is an example carried out by Felipe Oyarzún, Simon Guerrero, in collaboration with Andrew Quest and myself, in which they included curcumin in a nanoemulsion, as I mentioned before. They used this nanoemulsion to prevent tumor reincidence and metastasis. Here we have a model developed by Andrew Quest, in which he injected the cells, B16F10 melanoma cells, to favor the growth of the tumor. And after several days, if they excise the tumor and suture the wound, without treatment, there is a tumor reincidence and metastasis. With Felipe, they administered the nanoemulsion at this place in the wound, and after that, suture the wound and wait several days. They observed that the absence of metastasis and reincidence. This is the model in which the nanoemulsion was administered here. As we can see here, in the control experiments, there is not there is recidivism and also metastasis in most of the animals. And after the treatment with the nanoemulsion and curcumin, there is not formation of tumors and metastasis. We attribute this effect to the stability in vivo that can be achieved with the nanoemulsion. Here we have in vivo images. After 15 days, it's possible to see yet the fluorescence of curcumin that is stable for several days and allow to produce the effect in the animals. We are working now, we're starting now in collaboration with Cristian Torres and Carolina Valenzuela, with Felipe Oyarzún and Andrew Quest, work, a clinical trial in dogs for sarcoma treatment. We wait also to translate this to clinical in human. It's important to mention also that the nano formulation works very well in different tumor cells. We will continue with other applications of our nanoparticles in the center. We are working in collaboration with Andrew Quest and Alejandro Corbalán in the use of exosomes for natural targeting of tumors. As I mentioned, exosomes exhibit organ tropism and specific cells targeting also. And we can also modify the surface of the exosomes by conjugation with molecules, with peptides, for example, to improve the targeting, we can include in the exosomes the cargo, for example, nanoparticles or peptides or RNA, different kind of cargos to favor the targeting to cells and address the cargo. Here we have an example of this. In this example, we have exosomes of B16F10 cells. It was known before this work that these exosomes can exhibit tropism to the lung. In this case, we incubated the cells with nanoparticles and we produce the exosomes. In the exosomes are contained the gold nanoparticles. Here we can see that we incubate the exosomes with different cell lines, and we can observe that there is a higher incorporation of the exosomes in B16F10 cells, the same cells that produce the exosomes. The exosomes exhibit tropism to the cells that produce them. We have here the characterization of exosomes. Here we can see cryotim with the nanoparticles, and we inject the exosomes in this model. In this model, the cells were injected by the tail vein, obtaining the metastasis in the lung. And after 19 days, we injected the exosomes, and we evaluated the biodistribution of the exosomes by fluorescence and also by computerized tomography. We can see the accumulation of the exosomes with nanoparticles in the lungs. On the other hand, we evaluated the gold concentration in the lung and in the tumor. We can see higher accumulation of gold in the tumor, and also we can see a certain tropism of exosomes by fluorescence in the lung and the tumors. We also evaluated the accumulation, ex vivo accumulation of the nanoparticles contained in the exosomes, and we can see in the lung, in the metastatic tissue, the accumulation of gold nanoparticles. In contrast with the animals injected with the nanoparticles without the exosomes, we observe the distribution of the nanoparticles in different parts of the tissues. We are now working in the in vivo detection of exosomes by imaging methodologies, and also in the use of exosomes labeled with gold nanoparticles for detection and for phototherapy. It means theranostics. Last, I will finish with the development of exoliposomes, are liposomes that contains a composition of lipids similar to exosomes, are synthetic. But the idea of this is that one of the problems that exhibit most of the nanomaterials is the biodegradability. In this context, we are working with, we develop this system in which we have liposomes, and we cap the liposomes with gold nanoclusters, small nanoparticles that form a layer of gold here. This material can absorb energy at the near infrared region. In the near infrared region, it's possible to cross the tissues. It's easier to cross the tissues, and we radiate with near infrared, and we can produce the release of the drug. After that, it's possible to destroy all the structure and it's easier to eliminate from the body. We proved this in collaboration with Sergio Lavandero for cardiac targeting, and we include in the exosomes angiotensin 1-9. Angiotensin 1-9 is a natural peptide that was proposed by Sergio Lavandero and Maria Paso Carranza to be used for the treatment of hypertension. On the other hand, Sergio Lavandero and Andrew West also have studied the use of angiotensin 1-9 to inhibit metastasis. For this, it's interesting to explore the formulation of this peptide. This peptide has a half-life time in the body very low, 20 seconds. For that, it's important to improve drug delivery. Here we have the nanosystem complete with the capped with the gold, and after irradiation with to produce the controlled release of the angiotensin 1-9. After irradiation, it's possible to disaggregate all the system and to have small particles. We proved this in a Langendorf system in which we perfuse the nanoparticles with to prove the targeting and also to produce to evaluate the effect of irradiation. Finally, as a conclusion, we can say that nanosystems allow the spatial and temporal release of drugs, for example, by using photothermal effect, and it's very clear that nanoparticles improve stability and delivery of anti-tumor drugs as curcumin, and we are using exosomes for target delivery and for imaging of metastasis. The idea also is to obtain nanoparticles, biodegradable nanoparticles that allow theranostic purposes. Here we have the people that participate in this work in red, Felipe, Eileen, and Ana Riveros, and students that were involved in this, in the results that I show you. People in which, with which we collaborate from different centers, and national collaborations with Andrew West, Alejandro Corbalán, Cristian Torres, Carolina, Lorena Lobos also. Thank you for your attention. Okay. We will continue now with Nelson.
Wait.
Nelson?
Yes. I'm looking for my presentation. Oh, okay, my presentation is someplace. Okay. Let me check here. I think it is. Just a second. That's all right. Okay. Now.
You have to share the screen.
Yeah, that's now in there, share. And put it up. Okay. Okay, that's it.
Okay, perfect.
I need the first one. Okay, that's the third one. Okay, thank you, Marcelo. I'm going to talk about our one part of our research in Brazil. So this is nanomedicine applied to the preclinical and clinical animal model. I have to clarify what the preclinical, which animal-induced cancer, and in the clinical animal, as you, Marcelo said before, is the clinical with dogs, is clinical treatment, it's not experimental. So the idea of this presentation is to show the bladder cancer in animals, how is the progress to go in after, this when a case is 24-fold doses, we have a total regression of the tumor. So this is going to, is related to my presentation. So we have, for a long time, we're working with a new nanostructure. This is the Oncothera, and this is oncology therapy adjuvant, okay? And in the term, this is for human treatment. And in the case of the veterinary application, we use a different formulation that we call Oncovetter. So this is related to the Oncovetter, although are the same active. So the synthesis of this compound is straightforward. We synthesize an inorganic complex by technique for nanostructure synthesis. We have a 310 nanometers roughly, the size, and associated in the protein, which is a natural source. And we have, in association of both, we have the Oncothera here with the roughly 400 nanometers. Okay, so even you can see here is the microscopy shows a very spherical form, very homogeneous. And then when you have the proteins involved, it's a little bit higher in the size, but still totally spherical. So we have a lot of patent with this compound for protecting, and we have in Brazil, of course, 13 countries in Europe and United States too. Basic is to know exactly what is the compound and how is the characterize this. Well, we use all our technique in nanostructure because we have a lot of experience for many years. So we have an NMR spectroscopy, also X-ray diffraction pattern, or also the X-ray fluorescence for determination analysis to the structural relationship, the mineral. And we have a FETR size and so on, Raman spectroscopy, and also, of course, the interaction of the inorganic part with the organic part. So by the circular dichroism, we know exactly what happened in this compound. So, of course, for using in animals, we have to know stability. So we follow all resolution the ANVISA. This is equivalent to the FDA in the United States. So we have a storage condition, long-term study of follow-up 25 to 60 degrees, accelerated stability. Also, we use this. So we are showing that very stable for 6 months and a room temperature. Also, it's important the validation of analytical method for this characterization. So we follow the rules of NanoRAC. This is European Nanotechnology Network, which is an agreement with INMETRO that is belong to the government of Brazil. So because we don't have rules for nanostructure in Brazil, so we follow these validations. Also, the bioavailability of Oncovetter was carried out in rats. Of course, the protocol used in animal was approved by the ethical committee in our university. So we studied the distribution of this drug in the rats or in the animals. Probably is the enteropathic reabsorption of this compound, and it's eliminated via or metabolism or simply by biliary duct. We saw it accumulation mainly in the pancreas and the spleen. Importantly, this is important that in 48 hours was totally metabolized or eliminated. This is the graphic that the concentration in the blood. So you can see you have 2 peaks, but the different concentration. So 1 at 1.6 hour, and then probably 2 hours of you have the maximum absorption in the blood, and then in the plasmatic, and goes elimination in 48 hours. Here you can see that I just told before that you have in case with pancreas, the concentration is in 1 hour is very high. So This is mainly one of the target of this drug, and also with the spleen in 1 hour, 2 hours period. So we know that is very related to our result that we observed with cancer in rats and mice and rabbits. Okay, so the preclinical study was started all the time with the cytotoxicity, but for us it's not very interesting, since the cytotoxicity is belong to some chemical or chemotherapy that is toxic to the cell, cancer cell. In our, it's immunomodulator, so it's not effect on as a cytotoxic. That is shown here in the bladder carcinoma set line 5637. So you can see no insignificant effect, so no cytotoxic. Also, we are following the micronuclear assay to see its genotoxicity. So we found out that the rats, mice, and also in rabbits, we can see that no effect at all. They are very similar to even a high concentration. We have three concentrations. You see is 20, 50, 100 milligram by kilogram in the rat. So no different with control, indicating that oncothera was not genotoxic to this dose study. These toxicological and biochemical parameters also we follow with the three animals, and of course the different enzymes and urea and creatinine in the three cases. Okay. Also we didn't see any changes even in a high concentration, 100 milligram kilogram. So did not differ statistically from their respective control, indicating that this compound did not present systemic toxic effect. Of course, it's important in terms of the immunomodulation is the inflammation effect on this drug. So we're around the same animal, all this experiment, and then I show you in this table only with the rats because they're very similar. And we observe that in the rats, the drug 100, the higher concentration, has intense inflammation in the urinary bladder, ureter, and kidney, 60 to 80% of animal. In mice, in that concentration has just moderate inflammation, but has it in roughly in the same organs on 100% of the cases. In the case of the rabbit, in the high concentration group, has moderate inflammation in urinary bladder and kidneys alone. Okay, so this is important thing that is. This is justified quite well why we can use in animal the process as an immunomodulator, because they get induce inflammation. So, and also the important that it's not toxic. So both things are interesting to study in animal. So the preclinical induction and treatment of non-muscle invasive bladder cancer with RAD344 feature. Just to remember you that the bladder cancer staging is this in this slide, that is have its different stages, and non-muscular invasive bladder cancer is T1S. This is in situ tumor. This is just in the touch the mucosa with the layer, and then very slow lamina propria invasion, but it's very simple, very, very, very soft. One time you go to T2, T3, T4, you have to eliminate the bladder, because it's the only way to treat. In this case, you have the chemotherapy and immunotherapy. That is the classical or gold method is the BCG, onco-BCG. That is the Bacillus calmette-guerin. That's the tuberculosis vaccine. So in that case, our induction of the animal should be only in this stage. So we use a cystography and ultrasonography to follow our urinary bladder. This is a normal rat. You see here just nothing, and this is air, just air. And you see here with the contrast injection, so you can see neatly the bladder, okay, in cytography. So it's very good for following this process. And in ultrasonography, we can see clean bladder. So it's very good. One time we induced the cancer with the NMU. This is the N-methyl nitrosourea. Okay, that is very, very good for inducing bladder cancer. So we have also the same thing with the cytography. We can see all the effect here. This is nothing. There's a contrast. We can see better. And then when we see, in this case, we can see mass infiltrating the ventral, dorsal, and cranial bladder wall. It's this asterisk, okay? And the of course, the also we see some reflux in this vesico-urethral unilateral. So it's neatly is related to our tumor that we are studying. Here is the ultrasonography. You can see neatly all the tumor here, okay? So it's very good. So the monitoring of urinary bladder cancer by ultrasonography on rats this is a normal. You can see, I don't know, it's clear, but here is nothing too. And in different conditions and different times, you can see the mass of tumor mass here. And just for checking that this is a real, it's not an artifact, we ran color Doppler mapping, showing intense blood flow within the tumor masses, reinforcing the diagnosis of urothelial neoplasia. So we started with the induction of treatment in rats. So the first is histopathology analysis. And this is summarized all our results. So we have this is the histopathology effect here and the control. This is just the carcinogenic, and then the carcinogenic and only the scaffold, the inorganic scaffold, and then is the protein alone. And also here with the oncotherapt or oncovector, depending on what the solutions. Or in the case of normal, you see 100% force, and the carcinogenic produce a typical phase of this non-muscular invasive cancer. So you have a flat hyperplasia, 20%. I mean, excuse me. I have a non-invasive papillaris, TTA, okay? And in 40% and 60% PT1. So this is a typical non-invasive bladder cancer. Okay, when we use the only the inorganic part, it was a surprise that it worked like immunomodulation. You know, 20% of normal, and flat hyperplasia that is benign is 20%, and of course, it not totally support the system that we have a PTS and then we have also PT1, okay? So also the protein has the same thing, 20% normal, 20% flat hyperplasia. So it's quite good and interesting because either, of course, that is supported totally the animals, so you gave it some progression of the cancer. But look at that in the oncotherapy. This is the 40 and almost 80% is into normal and flat hyperplasia. So it's recovering almost totally. And very small, 20% of the rats has some PT1, it's a tumor in situ. So here, this is corroborated by this technique. The immunochemistry, in order to understand a little bit what happened with this, we follow the some of these markers or proteins that they probably belong to this mechanism. So the toll-like receptor 4, you see here in a normal is 75% the rats and 54 in the case down in the MNU, that the carcinogenic, and then it's recovered up to 71%. Where is my oops. Okay, 71%. So untrift, one of the cascade in the immunomodulation, as you talk a lot about this morning about this, all this stuff. So we have also the normal has a 80%, 82%, and then you have 71 recovering, so the cancer disease. Interferon gamma is extremely induced. You see 85% of the rats here in the cancer is 53 and 50% in the control. Also important thing here is the relation, the rank L, which was inhibited in our presence of oncotherapy that is induced in MNU, that means metastasis and progression of the metastasis. So oncotherapy inhibit this process. So we use different kind of mice in order to understand what is important in this, and we use the wild type mice, and you see here is quite nice the effect of the oncotherapy. And we compare with BCG. Remember, BCG is the gold system, gold treatment in bladder cancer. Okay, so it's okay, but it's 20% on the normal, and then you have other stages here, and not the resection and so on, and the progression of the cancer is not totally controlled. But in our case, it's okay. When we use a knockdown with the toll-like receptor 4, we have some effect here in the oncotherapy, but it's not totally effective. So we're up here with the PTA in this situation. So what happened? That probably the this oncotherapy in the toll-like receptor 2 is working on that now. So the deviation of the toll-like receptor 4 to toll-like receptor 2 that is not so efficient, but it's work. Okay, so this is interesting that the toll-like receptor 4 is very important for our drug. Even we used on a knockdown with the MyD88, which is a very important part of the mechanism. And of course, in the case of BCG, that is important too, because it's also by the receptor, the toll-like receptor 2 is important, this exam too. And we have no effect at all. So this is very important for us and in our mechanisms. Also, we're following the P53 that is important because the activation of the interferon pathway on oncotherapy or oncovetor was more effective in the treatment of this cancer in relation to the signal inflammatory cytokine mediated by BCG. So that oncotherapy or oncovetor can be considered as important therapeutic alternative of this type. You see here, in the wild type, you have a good induction of P53, but in the toll-like receptor knockdown is decreased. It's not so efficient on this, it's not decreased. But in case of MyD88, it's totally repressed. Okay, so no effect of oncotherapy. So this is we propose a mechanism. This is a preliminary mechanism that apparently is the activated innate immune system mediated by the toll-like 2, okay, toll-like 2 and toll-like 4. And this process results in the increase in interferon signaling pathway. You see TRF, IRF3, and then the interferon gamma. And then you goes on all these processes through the macrophage and natural killer cells, and also the cytotoxic T lymphocytes. So this is our most important pathway here. They are also is capable of decreasing the rank L system expression in animal model, on consequently of preventing metastasis, formation, and progression. So in the animals, veterinary cancer, we have a lot of study and with different kind of these, and this is the clinical part. So this mastocytoma, we have a good result, lymphoma, memory, and melanoma also. But I'm going to talk very rapidly with the bladder cancer. So what is our solution in this case is the application of the oncovetor and Immuno, okay? So we have excellent results in different veterinary cancers. The innovation in this is does not exist immunomodulator in veterinary cancer treatment and exhibiting non-toxicity in the world, okay? So this is very important. So I'm not talking about the bladder cancer in dark. The diagnosis is 20,000 dark in the United States for a year, and Brazil is 80,000. So this is good numbers. So the patient presentation, in this case, I just showing you 12, we have 80 right now. So the median age of presentation is 12 years and a half, so old dogs. The median weight here is 7.6, and the gender, we have female intact, female spayed, male intact, male castrated. In the breed, we have a lot of breed: Dachshund, mixed breed, Yorkshire Terrier, miniature poodle, etc. So it's very variable, the drug we are studying. So here a comparison of tumor volume of canine bladder before and after OncoVitor treatment is, you see here is before and then after Oncotherapy treatment. So 84 reduction in tumor volume, roughly. This is more than that because it's only 6 examples. You see, it's very, very efficient, the decreasing of this blood tumor volume. So this is to show that this table showed up that the volume before is 10.7 curie centimeter, and then goes in the 6th administration as 4, and then in the 25 is 1.1. So this represent at the 24 administration 84 or 90% of regression. So this is to show you that when we follow the animal, this is the tomography. So we see we have to see here the tumor. Okay, this is the bladder, and look at that in tumor, and then it's this no dose, and then the second dose or first dose here, and then it goes in 3 and this third dose, and the 6th dose, and 18 and 24. So you have a totally regression practically of the tumor. Okay, so also besides this observation here, hematuria is a presence of red blood cell in urine. It's an important clinical sign. Okay, of bladder cancer recurrence or progression, and OncoVitor treatment significantly reduced hematuria after the last application. This is directly related to the partial remission and tumor observed. Okay, look at that, the average survival free of disease with or without OncoVitor or the other one. The canine bladder cancer should be here. OncoVitor, without OncoVitor, 161, and with OncoVitor, it's 540 days. So the increase of surviving is tremendous. So this is the our 2 compounds, and this is our group. Thank you.
Okay, thank you, Nelson. I want to apologize because I didn't present you. I will present to you, and now to Wagner. You will give the Wagner talk, but sorry about that. I was...
No problem. No problem.
Okay. A moment, please. Okay. First of all, Nelson Duran, our last speaker, is professor of biochemistry at the Institute of Biology, professor of chemistry at the University of Campinas, and actually is a professor, invited professor at Unicamp, at the Department of Structural and Functional Biology, and coordinator of urogenital carcinogenesis and immunotherapy laboratory. And at the nanomedicine unit at the Federal University of ABC, Brazil. He is licentiate in chemistry in 1977 at the Universidad Católica de Valparaíso. He received his PhD in 1972 in University of Puerto Rico and a postdoctorate in Sao Paulo. His present research interest is oriented to nanomedicine and nanobiotechnology in the field of oncology. He is responsible for the synthesis, characterization, and production of Oncotreat, the immunomodulator that we heard recently. He is a member of the NanoReg, a common European approach to the regulatory testing of manufactured nanomaterials. Sorry, Nelson. Now, Nelson will give the talk instead of Wagner Favaro. Wagner Favaro is professor of anatomy, pathology, and oncology at the University of Campinas. He received his master, PhD, and postdoctorate in Unicamp at the same university. Since 2012, he is assistant professor at the Department of Structural and Functional Biology and coordinator of urogenital carcinogenesis and immunotherapy laboratory at Unicamp. He is a full member of the American Society of Clinical Oncology. His present research is oriented to uropathology, carcinogenesis, urogenital tumors, immunotherapy, regenerative medicine, and nanobiotechnology in oncology. He was the responsible for the development of Oncotreat, which has therapeutic application in several tumors. He also is a member of NanoReg. Thank you. Please, Nelson, if you...
Okay. Thank you for the presentation, both. But actually, I apologize for the absence of Wagner because he has a big problem. But anyway, I'm going to try to show you that the results. So let me share your. Okay, this is sure. That's okay. That's okay?
Yes, perfect, right? Nelson.
Okay.
Yes.
Okay, thank you. Thank you, Marcelo, for the introduction of Wagner. So if you have hard questions, please send this mail to him because this is related exactly to this area. So, but anyway, I know a little bit. It's not so bad. Okay, so the phase 1/2 clinical trial of innovative nano-immunotherapy in bladder is our bladder cancer, is this topic. So now we're passed through the clinical and preclinical in animals to go to the human treatment. So this is the topic. So why we are interested in that the significant unmet medical need in this cancer is very big. We have roughly 460,000, 430,000 new cases in the world for this kind of cancer. And this is curious because 70 to 85% is in non-muscular invasive bladder cancer, okay? Bladder cancer is the single most expensive in the United States. It's almost 4 billion by year. So it's interesting to study this kind of blood, this cancer. One of the worst patient experience among common cancers, say all the patients, okay? So we have a problem with BCG shortage. This is complicating patient care, okay? So in Brazil disappear from December, November until March or April, we have no BCG or onco-BCG for this cancer treatment. So it was very, very, very bad. Survival rate for bladder cancer have decreased in recent years in the several countries, during which time there was also a BCG shortage, as I said before. What happened with after BCG in patients, option is to, when it doesn't work, is undergo radical cystectomy or phase cancer progression. This is mean to eliminate in the first cycles or second cycle with BCG doesn't work, you have to be a cystectomy. That's the rule or the recommendations. So what the radical cystectomy remains recommended treatment option after failure, okay? well, in the 6 to 7% lifetime risk of the cystectomy associated with the and then this thing. So this is a risk. Long complex surgical procedure. This is high hours. Significant rate of morbidity and 60 to 30 to 10% within 19 days. And mortality is 2 to 9% within 6 months. 64% complication rate with 90 days. 35% of patients require an emergency room visits, and many cases, 26%, require readmission. So additional complication can occur, and most patients with this cancer are elderly, yeah, and often with comorbidity. So tremendous impact to patient quality of life. Life following radical cystectomy require catheterization and urinary diversion, so it's not easy. Well, our proposal is a new immunotherapy to this solid tumor treatment. As I said before in my presentation, yes, it's a nanostructure, inorganic complex associated with the glycosidic protein and developed in our university, which exhibit immunomodulatory and anti-tumor properties. We have a patent in Brazil, and we have in 13 countries in Europe, and recently in the United States. So we are quite protected from other studies or institutions. So the synthesis, as I described before, very straightforward. So it's interaction, the synthesis of this inorganic scaffold is relatively simple. You have the 310 nanometers, and then you interact with the protein and then associate it quite nice to produce the oncothread as described before. Also, the mechanism is the same in human and in the animals. So the proposal for immunomodulation is quite clean. What kind of patient we are using in this scan? Because our compound is not until it's approved by Anvisa, neither FDA, right? So we have certain conditions for the patient when they have no hoplets with that. Okay, so one time you have rejecting the BCG or onco-BCG treatment, you have to cystectomy. So many of these patients that doesn't want to do that, so enter in our clinical trials. So it's compassionate protocol. Okay, so in that condition we can use it. And also is registered by the Ethical University and also by the government in the clinical, say, like a very equivalent to the clinical trials in government in the United States. So what is study? It's a design, our design. Okay, it's prospective single center, only one center. So single arm phase 1/2 study with 29 patients. Of course, the 29 patients are very, very close to, is following by the trials, and it's very excited in terms of the following of this. We have more than 180 patients right now in different type of cancers, but this is approved by just for this kind of bladder cancer in our university. So, and we have an association with the Paulina Hospital. Okay, so we are 18 males and 11 female, all with high grade non-invasive molecular bladder cancer. So it's refractory or relapses, okay, to the BCG treatment. At least they have one previous course of BCG intravesical therapy. We can apply in any patient that doesn't pass this kind of condition. The schedule was initiated with weekly intravesical, 120 milligram per ml, and intramuscular, 25 milligram by ml, and the treatment for 6 weeks, followed with one every other week's application for 3 months, and 1 month application until the end of the treatment in 24 months. Follow-up was performed with systematic mapping biopsies of the bladder and cystoscopy and ultrasound. Okay. The primary endpoint were pathological complete response, PCR, and recurrence-free survival, RFS. Right? The recurrence was defined as histology proven tumor recurrence, any grade, and monitored at 3 months intervals. Secondary endpoint were time to disease recurrence safety response. So that is our distribution of cancer and of the stages of this histopathology grade in phase 1, 2. We have a 10% of T1 PTS in situ, PT1 31%, and PTA 59%. So this is the distribution of our patients. We have a frequency category variable as different kind of situation, as biological grades and histological grades. Also, the numbers of relapses, of the tumor focality, and also the complete response and recurrence. So here we have a summary of one of the samples with from relapses. We have in the 3 to 9 months, we have no relapses at all. Okay. In the 10 to 13 months, we have 3 cases. What it is counted for this, that means 16%. And also in the in the 10 to 12, we have 3 persons. Okay, and in 13 to 1, and then in 12 and 12, 2. Okay, so the recurrence is not very usual. The histological grade, we have a high grade PT1, 2 patients, and PT1, PTA 1, and PTA 3. So this is the percentage of this. The tumor focality is a multifocal, only 1, so many tumors in the bladder, and a single was on almost all these patients, 83% of them. The tumor size, 100% were less than 3 centimeters. So this is important. The complete response was almost 80% was complete response CR, and recurrence-free survival was 100% in 24 months. The evaluation, the focality before BCG and after oncotherapy. Okay, you have BCG is a high grade in the case of histological grade, is 100% of the patients, and we have a distribution in the high grade 50% and 50% of low grade. Okay, and tumor size in the case of BCG is 10.3%, is lower than 3 centimeter, and higher than centimeter is almost 90%. In our case, is that the higher at 83% is the less than 3 centimeter, and then that. Tumor focality was 41.4 in the multifocal, and single 50 or 60%. In our case is 16.1 in multifocal, and or single focal is on 83%. So we know exactly what happened with these patients. Okay, so look at this important thing in all the other patients is the number of recurrence before and after oncotherapy treatment. Okay, of course this is the treatment with onco BCG or intravesical chemotherapy. Okay, this is the number of relapses, very high. And look at after that in the 3-9 month, nothing. And then you have a 10 to 12 very low, and lower in 3-13, and similar to the 19. So the effect is very great. Pathological complete response result in our phase 1/2 clinical trial. We have this is the time point, and evaluate patient is in 3 months, 100%, 6 months, 100%, 99, 100%, 12 months, we have a 90%, okay? And in 12 months, we have 80% of pathological complete response. Recurrence-free survival was the 22 month after 24 months follow-up, okay? The median time to disease recurrence is 618 days. So this is a very long period. And duration of response is 50.3 months. You can see here this and this recurrence-free survival, okay? So is very impressed. One thing we find out in all similar cases, not in all, but in the most patients, is 52% in the thread form. We find out that this immunotherapy, importance of this appear lymphoid follicles, primary lymphoid follicles, and the tissue. So we are analyze this immunohistochemical reaction to see what happened and what is the importance of this case. This primary lymphoid follicle form network of follicular dendritic cell, as just was explained before, which is important in cancer immuno system, which are important for geminal center reaction, during which affinity maturation creates optimized antibody in adaptive immune response. The toll-like receptor 4 is involved in the maturation of this follicular dendritic cell and in the development of the germinal center in the lymphoid follicles. Improved antigen presenting cell function as the release of cytokine and cytokine essential for the activation of CD8 and CD4 T cells. Okay, so this is very important effect in our treatment. Also, the semi-quantitative analysis, the monolabeled antigens in this before and after oncotherapy, we have also very similar in the result in animals. The toll-like receptor 2 was induced, and the toll-like 4 is induced too. So the TRIF is a tremendous effect before oncotherapy treatment and after treatment. Gamma is from interferon gamma from 121 to 25 of the cases. So and also we studied the induction of nitro oxide synthase. Okay, also is important in this mechanism. One time we have adverse results, we have to know exactly what happened with the adverse reaction. This is very important in clinical trial. Okay, so we are differentiated in 1-2 grades and 3-4 grades. Oops. So the total 22 patients exhibited almost 75.9% adverse reaction resulting from the oncotherapy, okay, this treatment, and 7 patients, 25% had no adverse at all. Okay, the grade 1-2 adverse reaction were significantly more frequent in the than grade 3-4 reaction. You see in this table, you see this is more populated and this is not. And a total of 18 patients, 62% had only grade 1-2, and the 4 patients, 14 had both grade 1-2 and 3-4. Okay, the most common grade adverse reaction is roughly 20%, where dysuria is 52%, pruritus in 44, 45, cystitis 34, and fatigue 27, and rash 75%. The most common grade in 3-4 adverse reaction was less than 2%, diarrhea, abdominal pain, skin rash, cough, and dyspnea. But all these conditions were totally reversed with medical treatment. So the conclusion is the oncotherapy, nanoimmunotherapy seems a safe and effective treatment option for BCG unresponsive, non-muscle invasive bladder cancer patients, and may provide benefit for preventing tumor recurrence. Also, this study demonstrates an important effect of activation of the toll-like receptor 4 signaling pathway, triggered by the oncotherapy in the formation and organization of this primary lymphocyte follicle, which may be related to the anti-tumor and immunoprotective effect from this immunotherapy in bladder tissues. So this is our boss. So Wagner is in this. This is Wagner, and this is our director in the uro-oncology laboratory. Thank you.
Okay, thank you, Nelson. Okay, we will continue with Silke. Andrew will introduce Silke.
Okay, Marcelo, I'll take over from here. So our next speaker is Dr. Silke Kroll. Silke trained in chemistry at the Westfälische Wilhelms University in Münster, and she got her degree there in chemistry in 1997. She then went on to do get her PhD at the same institution in 2000, continued there working for another year or so as a postdoc, and then went on to Italy to work at the Istituto Nazionale di Fisica Materia, so the Institute of Physics and University of Genoa, for about 4 more years until 2007. Then she decided to become independent, and between 2007 and 2010 she established and headed the nanobio lab so for nanoanalysis, drug delivery and diagnostics at CVM in Trieste, Italy. Also from 2010 to 2018, she then headed the laboratory of nanomedicine at the National Institute for Neurology, Carlo Besta, in Milan, Italy. She extended this work and intensified the work by going into the area of liquid biopsy for early diagnosis and advanced therapy on cancer, working at the Institute of Oncology, Giovanni Baoli II, at the Research Hospital in Bari, Italy. That was between 2016 and 2018. And since 2018, Silke is working with the IRCCS. Let's see if I can get the pronunciation right here. Ospedaliero Specializzato in Gastroenterologia Severo de Bellis. And there she's developing a novel nanoparticle-based therapeutic and diagnostic approach for inflammatory bowel diseases. But I think what she's going to talk about today is some of her work. She was actually a discovery of what are referred to as spheroid-inducing properties of inorganic fractals, and so that's what we're going to hear about today. So thanks for joining us, Silke.
Okay, thank you very much for the invitation. Thank you very much for the introduction. And you see the advantage in working on nano and bio. You can do a lot of different things. You can cover a lot of different diseases. And actually, I will go, let's say, back a little bit to more basic research, while the former talk was extremely applied and already very advanced. I am trying to introduce a new system, let's say, for an improved preclinical testing in order to reduce perhaps even animal experiments. And it's work we started something like 2 years ago in collaboration with the University of Twente. And it's okay. Okay, I have to start with a disclaimer because we filed a patent on the technology, and we plan to commercialize the cell growth platform in the future. And so for that reason, I want to disclaim that there's a conflict of interest. So at the moment, current medicine is that one treatment fits all. In most of the cases, the therapy somehow works. Then in some patients, there are no effects, and in some patients, fewer patients, luckily, there are adverse effects. But we don't have any possibility, let's say, really to tailor treatment to the specific patients. And we always talk about that we plan to develop, let's say, the personalized medicine, that future medicine is personalized medicine. And so far, what we do already is that we, let's say, define certain patient groups which can be treated in a particular way basing on their genetic setup. But let's say at the end of the day, we are not able really to provide a personalized treatment for one patient because we have a limited number of drugs. We have some experience what could be, let's say, the maximum tolerated dose in most of the patients when a drug is tested and approved. And let's say what we want is perhaps a fast therapeutic decision-making, individualized treatments, in particular for tumors like rare tumors where no, let's say, defined treatment option is available, where perhaps we have to test different combinations of different drugs to see how this individual tumor is responding. So how can we achieve really personalized medicine, for example, in cancer or in particular cancer? What we need is a piece of the patient's tumor, so primary tissue. Then what we want in order to, let's say, make a relatively quick decision is that this tumor tissue is let's say, in a lot of different organoids growing in a very short time. These organoids made of, let's say, several primary cells, all of them from the patient in combination, let's say, with their neighboring cells, allows that we have models which are mimicking the, let's say, the reality. And in this case, we have the possibility, if we have a lot of them, that we can test, for example, in different templates, a lot of different combinations of approved drugs to find, let's say, a mixture of drugs which are, let's say, to which the tumor is best responding, meaning that we have the best efficiency. So far, let's say there are several techniques which allow either to grow organoids or spheroids. There are actually two main strategies. One is that I just prevent that cells are attaching to surfaces. So either I use non-adhesive plates with a specific medium, or I use hanging drops, in which I, let's say, have a drop in which I have a cell suspension, and due to the fact that the cells cannot attach to any surface, they are attaching to each other and form spheroids. Or I keep the cells in continuous movement for growing. Or I embed them in Matrigel, which is actually one of the standard techniques. All of these different techniques have advantages and disadvantages. For example, if we are going to the hanging drop cell culture, we have, let's say, okay, in 2D cell culture is the standard model, but we can all agree on that this is farthest from, let's say, reality, because it's a very artificial system in which a lot of the typical parameters like microenvironment, like vessels, like specific conditions are lacking. And so they are, let's say, the basic model, but they, let's say, even for testing drugs and drug response, we know that, let's say, the system is somehow too artificial, and all the other systems also have some advantages and disadvantages. Let's say the hanging drop culture is difficult to handle. I have just one microtumor, and for that reason, it's and a very, very small volume. The Matrigel embedment is something which needs a lot of time, because I, let's say, implement more or less single cells which have to grow to microtumors. And very often in this process, the microtumor properties and the mutations are changing. And the standard model, especially for rare tumors, is that I have xenografts, which are implanted in immunodeficient mice. And this is on the one hand very time consuming because it needs several months before I can start testing different treatment options on these animals. And on the other hand, the European Commission now tries to reduce number of animal experiments and tries to look for a replacement. And in this particular case, let's say we found our model, which is actually somehow something between it's not embedded, but it's immobilized, so it's not hanging drop culture. You will see in a second. So what we have is inorganic fractal structures made of amorphous silica. And as you could see, we have two different orientations, a hexagonal one in the bottom and a square one. And wait a second, and a square one in the upper part. And we have different generations. So we have the most simple one, which is G0, which is, let's say, just a pattern of pyramids. Then we have the more complex G1. We have the even more complex corner lithography prepared G2, in which on each corner we have an additional, not pyramid, but route, whatever. Then we have G3, which is, let's say, growing again on all the corners, another structure. And then we have G4, in which, again, we have, let's say, structures on each corner of the G3. And as you could see, we get quite complex structures in the nanometer range. What we did was we tested this first with primary tumor cells. This was a project which we had at our institute, in which we were isolating actually cancer-associated fibroblasts and were studying them. And usually when you do cancer-associated fibroblasts and you isolate them, what you get obviously is not complete removal of tumor cells. And what we observed was that when we had a flat surface, you see the typical structure with the stress fibers and this very flat extended fibroblast structures. However, if we are going to G0, you see that on one hand, we have a cell layer in the bottom with the extended structures, as you could see also on the flat surface, but we observed frequently that we have three-dimensional microspheres, microtumors or spheroids on top of this. Or in this case, it's not spheroids, but it's most probable it's organoids of tumor tissue. This is different when we go to G1, G2, G3, and G4. In this case, we always get highly activated fibroblasts, but we don't get three-dimensional microtumors on top of a fibroblast structure. This is, let's say, the cell culture on day 13 from the calf cells isolated from hepatocarcinoma from a patient. So then we wanted to understand what we exactly see, if this could be, let's say, an aggregation or accumulation of some fibroblast cells, or if this is really a microtumor. So we tested this. Here you see the image of the microtumor with DAPI staining, and what you could observe is that we have nuclei on the outer layer, and inside we have a diffused DAPI signal, which is indicative that we have a necrotic core inside of an intact cell layer, as to be expected from a quite huge microtumor. And then we use the staining for alpha SMA. Alpha SMA is, let's say, indicative for fibroblasts, for calf cells. And as you could see, we have in the bottom layer, so the flat layer, 2D layer, let's say, directly attached to the fractals, we have a quite intense signal of fibers, which are the actin fibers. And around the tumor, more or less in the bottom, we see a very high intense signal, but it doesn't look like that we have something, let's say, in this outermost cell layer. And then we did staining with alpha-fetoprotein. Alpha-fetoprotein is a molecule which is expressed in tumor cells, but not in fibroblasts. And as you could see, we see a very dim signal in the bottom layer, but we have a high expression in this, let's say, three-dimensional spheroid structure on top of this 2D layer. And for that reason, we concluded that we have a microtumor on a cell layer of calf cells. Then, we tested at which time point we are starting to get these spheroids, because we tested initially on day 8 and we could already see them. Then we saw them on 13 days. But as you could see, we already see the first accumulation on G0, let's say on day 1, and then they are increasing in size and in density. But remember that this is not cell lines, this is tumor cells from the patient. Then we tested also cancer cell lines, and we did the comparison with the growth in matrigel. And here you see HLF. HLF is a cell line for hepatocarcinoma, a very aggressive one. And we compared the results on our surfaces and the size of the microtumors we observed with microtumors grown in matrigel. And as you could see here with the HLF cell line, we saw on day 4, let's say size more or less of around 75 plus minus 20 micron in diameter, while after 13 days in matrigel, so definitively much longer, we saw, let's say, an average size of 108 micron plus minus 50. So in this case, it looks like that the formation of organoids, of spheroids, of microtumors is much faster on the surface of the fractals in comparison to matrigel. And the advantage is that we have the microtumors immobilized, but they are accessible because they are not embedded in the gel, in a gel, but they are accessible more or less for all, from all sides. And then we tested also the growth of CACO2 cells. And here we have, after 8 days, we observed that the tumor size, the microtumor size, is already, let's say, in the range of 150 micron plus minus 50. So let's say it looks like that the formation and the growth of three-dimensional structures on the surfaces are definitively much faster as compared to matrigel. Then we tested the growth of human adipose-derived stem cells. And again, we tested, let's say, different surfaces in different configurations. And as you could see, also for the adipose stem cells, we observed that there is a formation of spheroids. but we were not sure what exactly it is. The only thing is that when you have a look to the structures, it already looks like that we have type of neuronal structures, and also these dense spheres look more than let's say like neurospheres rather than like accumulation of stem cells. So what we did was we were testing for neuron, let's say for indicator, either for neuronal stem cells like nestin. And as you could see in this image, we have the green signal for nestin, and you see that there is a nestin signal. We have NOI-N, which is an indicator which is for mature neurons, and they accumulate in the nucleus of mature neurons. And as you could see, due to the DAPI signal, we have no co-localization. You still see a very clear blue signal. We see some diffuse red signal, let's say more or less in the cell body, but not in the nucleus. However, if you are going to the situation changed. What we see is that the cells are no longer, let's say, in this neurosphere structure, but they are extended. They are, let's say, looking much more like neurons. And in fact, when we are testing with nestin, we still have a nestin signal, but what we clearly see is that we have a NOI-N signal, which is localized more or less in all nuclei. And you can conclude this because we have not a blue signal any longer, but we have a violet signal, which is due to the overlap of NOI-N and the DAPI signal. So for that reason, we were surprised because when we went then back to literature, we found that usually the differentiation from adipose stem cells, from adipose stem cells to neurospheres is usually quite fast. And it's usually in serum-free condition. We are not working in serum-free condition, but however, we get neurospheres very quickly. And then usually the differentiation from neurospheres into neurons is a very lengthy process. It needs several weeks before you are able to differentiate neurospheres into neurons, while we need 8 days. So this is let's say, the fast track to differentiate adipose stem cells into neurons and into neurospheres. And we expect that depending on the pattern and depending on the fractal structures, that most probable we can stimulate by that the differentiation perhaps also into other lineages. The other thing I have to mention is that we didn't add any differentiation factors. We didn't add any growth factors. We didn't use any stimuli in order to differentiate the cells. This was something which was triggered most probable by the interaction between the cells and the surface. So this is the team. This is Matteo and Francesco, who did the work. And initially I started this work with Simona. She was doing, let's say, the first work on both on the stem cells and also on some cancer cell lines. And then Matteo and Francesco were taking over. Francesco is our expert for CAF cell isolation. And then these are actually my partners in the new founded startup company. This is Arturo and Niels and Erwin, and we are starting, let's say, hopefully in December, this adventure in trying to commercialize, let's say, this surfaces. The company is Encytos, and as I said, we will start in December. Thank you for your attention.
Thank you, Silke. Thank you, Nelson. Sorry, I want to. I can open my video.
Me too.
Okay.
Okay. Yes. Thank you very much to Nelson, to Silke. Okay. Yes, I can. Sorry. Yeah. Sorry. Yes. Thank you to Nelson, to Silke. I want to congratulate to Nelson for the excellent results of the clinical trials. It's a big challenge here in South America. And also Silke, to you for your constant innovation in science. Congratulations. It's very interesting.
Thank you.
I have questions. I will start with the last for Silke. Is my question. How reproducible is the behavior of the cells in the different phases of the silica? Because you show in the top, okay.
You mean how reproducible it is on the different generations?
Yes.
It's completely reproducible. Let's say whenever we seed the cells, we always get microspheres or spheroids. So the size is, let's say, as you saw, we have a size distribution. They are not always homogeneous in size, but the growth of spheroids is absolutely reproducible. And always, let's say, when we have CAF cells with, let's say, some contaminating tumor cells, we always see on G0 micro tumors and not on the other ones. And when we do the adipose stem cells, we always see on generation 2, 3, and 4 that there are this differentiation into neurospheres and into neurons.
Okay, thank you. Other question for Silke. Do you think that from Lizette Layton, do you think that the signal to differentiate the neurospheres came from the mechanical stimuli generated by the fractal structure?
I think so, yes, because let's say the conditions, actually the conditions are always the same. What is different is, let's say, the distance between the structures, and this seems to indicate, let's say, induce the differentiation. What we didn't observe yet is, let's say, one of the other lineages, and we have to check if when we are changing the pattern, or let's say the complexity of the structure, if in this case we can induce a differentiation into, for example, osteoblasts.
Okay, thank you. To Nelson,
Yep.
I have a question from Lorena Oroztica. Thanks for the talk. Is this treatment functionalized? I mean, is the compound targeting, or could it specifically target the bladder? You target the bladder?
Yeah. No. It's not cytotoxic, so it's not the cells attack. I mean, all the indication of our mechanism in animals and also in humans, that is the activation of the innate immune system. So probably, but through this system is just this, is initiated all the process for the protection of the animals or the patients against this cancer. So I don't think so is this related cell directly like a cytotoxic cell. So in that case, you have to charge it, or surface charge or something to folic acid, for instance, and so on for cancer cell, but it is not our case.
Okay. I have two questions. One is my question related with adverse reactions. which is the cause of the adverse reactions? How you explain the adverse reactions? By which mechanism?
We don't know. We're studying that. We don't know exactly. That probably is, in all the immunomodulators that uses in now in the medicine is very similar effect. Okay, rash, allergic effect, and so diarrhea and all. So this is very common. I don't know exactly, but we are trying to study that to understand, because in dogs, for instance, we don't see so many effects. We don't see it. The only thing we see is allergic. Sometimes in some dogs, not all, 5% of the dog, it's exhibited a very, very strong allergic reaction. But then as we treat it, and no problem, none was died. But I think that it's a very common system. We don't know exactly what happened, but this is very common in immunomodulators.
Yes, Andrew's question was why in dogs you don't see the effect.
Yeah.
Yes.
We see some effect, yes, but not so strongly than human.
Okay. Yes, other question. You have a muco addition on the how stick the formulation to the bladder. How is retained?
Yeah. Probably some muco addition, because we inject intravesicular, right? So direct in the bladder. Okay. So probably there this is adhesive in the surface of the tumors. Yes, it's possible to have some. We didn't study that, but I think it's a reasonable explanation for that. Yes.
Okay. For Nelson, other question from Andrew. As you incubate your nanoparticles with proteins, they increase in size exceeding 400 nanometers. Have you noticed that size become limiting to therapeutic efficacy?
No, we didn't see any effect. You see, I show you the very similar activity with the scaffold, the inorganic part. So it's not I think it's not effect. I mean, we don't see any different effect on just the scaffold and also the oncotherapy. So no, we don't think it's important.
Okay, thank you. I don't have more questions here. Thank you for the speakers. We will finish this section. And now, Andrew.
I have some questions here. I wrote down, Marcelo.
Okay. Thank you.
There's a question for you from Silke, and she was asking how you actually load the exosomes with drugs.
Okay, sorry. Yes, I are very above in the tone. Yeah, sorry. In the results that I show, we incubate the cells with the gold nanoparticles, and the cells produce the exosomes containing the nanoparticles. Yes. On the other hand, we can incubate the exosomes with the curcumin, for example, or by electroporation. But the idea of electroporation is that you can modify the targeting after the treatment. Okay. Yes. There are questions for me also. Yes. Eduardo Duran, yes, asked to me. Yes, have you tried to load these nanovesicles with molecules such as integrin to improve tropism, or with therapeutic oligonucleotides? We didn't, but it's a good idea. There are reports about that, Eduardo. Yes. And Lorena also. Have you carried out toxicological or clearance evaluation of these nanoparticles in this animal? In the case of the work with Andrew, yes, they did toxicological studies, a very complete profile of toxicological studies, and they didn't see effects. In the case of other formulations, also not so complete like this toxicological profile. Other questions? Okay. Yes, no. There are no more questions. No. We finish now the section, Andrew? I don't see.
Basically, with this, we close the final session here, and we'll move on. So we created a little presentation with highlights of the meeting. And so we go and revise those. And I believe we're still not ready with the ones from today. So I'll start with some of the highlights we put together for the first few days. And then by the end, we should hopefully have the ones from today. Let me see if I can get this to show up here. Okay, I'll go into screen share mode now. Okay. So I should mention here that this was a joint effort between a number of different people, which will be mentioned at the end. So the idea was to summarize some of what we considered were the highlights of this meeting, starting from the first day, which was the 3rd of November, then the second day, the 10th of November, and afterwards we should have some highlights from today as well. Alejandro, are you there somewhere?
Yes, I'm here.
So we'll be commenting on these highlights together there. So I should also mention that these highlights were selected based on comments predominantly by some of the students who attended the different meetings. And then in the case that an individual person made this contribution, their name is mentioned at the bottom of the slide to the right. Sometimes it was a suggestion by several people, so we didn't necessarily mention everyone involved. So, but one of the first highlights that was mentioned is a presentation by Vicente Torres, and these phrases that show up here in the slide are basically taken straight from his presentation. So what he was saying is that endocytic pathways apparently promote migration in different ways, so migration of tumor cells. This involves assembly and disassembly of focal adhesions, Rab5-RAC1 activation, E-cadherin and beta-catenin degradation complex endocytosis, and finally destruction. So based on these observations, understanding endocytosis in cancer cells would seem to be an important clue in order to improve treatments directed towards avoiding metastatic processes. And then there was another comment regarding Vicente's presentation, so where he alluded to the possibility that some of these mechanisms might also be important in stimulating the migration of cells basically in the gums. And so he's saying here the components of the saliva are likely to, based on their findings, to promote migration of epithelial cells, again, by activating Rab5 and then downstream RAC1. So this seems to be a conserved mechanism then not only in cancer cell migration, but also in oral wound healing processes. This is for you, Alejandro.
Yes, this is Luis Carvajal, who presented here the high rate of mutation of gastric cancer, which is on the left-hand side. And the other side is something that's quite striking. Thanks, Maria Fernanda, for noticing this. Not all mutations are clonal, but it's showing here that with the concept of founder or clonal mutation and private mutation, is important in terms of precision medicine, since not evolution of the tumors is based on the founder mutation. So this is a key element to keep in the future, and is the explanation, the succeed or the failure of molecular therapies.
Okay, another one for you there, Alejandro.
Yes, this one is something that will be a continuum in molecular genetics in our countries. This is a comparison between TCGA, which is the standard, in which there are more than, I will say, up to 500 cases of gastric cancer that have a full molecular sequence, but only 60 already from Brazil, Latin American countries. And this is the personal effort of Luis Carvajal, that with collaborators from Mexico, they were able to put how it looks the TCA when it's done outside the TCA in Latin American population. And there are some novelties, but some things that are already known. So I think it's something that we keep following, because it's opened the door to identify our own mutations in our own tumors. And this is first effort.
Okay. This is also one for you.
Yes, this is also an effort from Gareth Owen to something that is written here is something that is not easy. Clinical data has been, of course, clinical medicine is defined by clinical information. But now we have a huge amount of molecular information that will contribute and reshape the clinical medicine. But integration is not easy. And this is an effort of Dr. Owen's group to integrate molecular profiling data to predict chemotherapy response, which is critical in terms of survival of the patients. So this is a Chilean, our own effort to produce that kind of approach, which is key in precision medicine.
Okay.
This is Dr. Arvind Sen also highlighting, I think, two issues. One is the presence of novel mutations, but also the capacity to work together different Latin American countries, which I think is something that we will have to put a major effort. As a country, we're a small country, and I think nobody sees us as a single country. People, the rest of the world, see us as a continent, as a region, Latin American region. So I think it's important to work in a cooperative way, especially in looking for novel things like Ricardo Armesen is doing. Well, this is my group, also showing same thing, but from hereditary type of tumor, which is important when you find a germline mutation. It's a founder mutation, and has some kind of ancestry. origin. So this is an example from a different type of tumor, which is the familial gastric cancer in this case, in which we have identified a germline mutation, which is novel, and also germline in non-coding genes, which open the door to non-coding genomics, which is a evolving topic in cancer research.
Okay, so we'll switch gears here and go back to more molecular, biochemical, molecular biology, with the presentation by Lorena Lobos, who is interested in identifying markers for basically early detection of breast cancer. And what she showed us is that lactidierin present in is actually is present on exosomes that are secreted by breast cancer cells, and this may serve precisely as an easily accessible marker for the early diagnosis of breast cancer. Okay, with that we now move on to, so that the ones we heard about now were the first day, so moving on to the talks from the second day, the 10th of November. And we had there a talk that was highlighted by Lisette Layton. She's working on studying mechanisms by which tumors undergo metastasis in this model using melanoma cells. And so what she found is that lung tumor mass can be reduced, very importantly, as you can see here to the left, when beta-3 integrin is silenced. This, together with other data she showed, supports the notion that metastasis in this particular system, at least, is due to an interaction between Thy1 and beta-3 integrin, which is an interaction she had actually previously described is important in the central nervous system between reactive astrocytes and neurons, and there's actually an interaction that precludes neurite extension and for instance, regeneration after damage to the central nervous system. So it's interesting that there are these parallels between things like the neurons, the central nervous system, and cancer cells and how they undergo metastasis. But of course, the interest here is also in that there are possibilities for therapeutically targeting these types of interactions. Then we had a talk by Denise Bravo that was also highlighted. She's studying the, let's say, effects of Helicobacter pylori, but not in the normal canonical context, which is of course in gastric cancer, or the development of gastric cancer, but rather how it can relate to oral health and development of disease states there, like for instance, periodontitis. So as pointed out here, beside the well-known gastrointestinal effects of Helicobacter pylori, there is evidence that it can also affect the oral periodontal microbiota, specifically by interacting with Porphyromonas gingivalis and increasing its virulence. So what she showed is that essentially cells incubated with the bacteria Porphyromonas gingivalis, they migrate more. And if they were co-incubated with the Porphyromonas that was pre-treated with Helicobacter pylori, this effect is actually great, as you can see here to the left in the panel there. And this effect depends on the expression, enhanced expression of proteases, cell surface proteases of the bacteria that are called gingipanes. This relates to the same presentation. Again, the connection between pylori and gingivalis, and basically showing that there is not only an increase in migration, but there is also an increased stimulation of production of pro-inflammatory cytokines. So there is increased inflammation in the basically oral microenvironment due to the interaction of these two bacteria. Then we had a presentation by Manuel Valenzuela, who's interested in, essentially based on previous studies, the interest there was to find out which factor of Helicobacter pylori was involved in the induction of hypoxia-induced factor, which is, of course, important for processes like angiogenesis, as we all know, and then also subsequently for metastasis. And what he saw or found is that the virulence factor involved in Helicobacter pylori that is responsible for the induction of HIF-1 alpha is actually a well-known virulence factor that had been previously described as essential for survival of the bacteria in the stomach environment, which is urease. And essentially what he reported is that this urease is actually not only an enzyme which can neutralize the acidic environment, but it's actually also a ligand which can interact with the toll-like 2 receptor and trigger sort of pro-inflammatory responses. And the interesting thing here too is also that this is actually present then on vesicles that are similar to exosomes that are outer membrane vesicles of these bacteria. So this raises the specter that actually these factors that are present on these vesicles, which can induce inflammation, will not only do so in a local manner, but can also potentially do so in a systemic fashion. Do you want to comment on this, Alejandro? Alejandro?
Yes, sorry. Well, this is a plenary lecture presented by Wael Refai. I think was a good example of translational research because he used, he trying to address a clinical question using in vitro mouse models, clinical samples, trying to characterize one specific gene, which is TFF1, which has a role in the precursor lesions, not only in cancer, and targeting STAT3 and NF-κB. And these slides show the expression of this gene in between normal. IM means intestinal metaplasia and dysplasia, which are well-defined precancerous lesions. So this is a good example of translational research.
Okay. So this was a presentation by Marcelo Hermoso, who reported on elevated inflammatory alarmin expression. So this is would be IL-33, and then stimulation of the IL-33 ST2 axis, particularly in colorectal cancer tumors, and this was associated by her with increased lymph node metastasis. And also she suggested altered expression of these molecules in inflammatory bowel disease and colorectal cancer identifies this signaling axis as a potential therapeutic target, as well as potentially relevant biomarkers that, of course, could be exploited in the monitoring of patients and controlling patients as they go through therapy. Alejandro?
Okay, well, this was the presentation of Sebastián Mondaca, which is it's like to look in the future. He did his training at a very top one, probably, cancer center in the world, so they have their own program for precision medicine, which would try to identify specific targets that can be druggable for these patients. And he presented several examples that not all of them have a clinical impact, but I think it's a way to move the oncology, the practice of oncology, and also for my view, I think it's important how today in clinical medicine you cannot understand how to manage patients if you don't understand genetics, and in between is the molecular pharmacology, which at the end is kind of cell signaling. So I think this is emerging topic, and particularly I push the idea to invite Sebastian, who is a young physician. I think his approach has a lot of future in terms of combining basic and clinical research, and will be ideal if we can establish these kind of programs here in Chile in order to be part of this movement in terms of understand the molecular basis of cancer.
Okay. Another one for you.
Well, this is an example of whole history that came true. Hernán González has been and also a physician who has scientific training, and he identified a clinical needs for thyroid cancer, which is not a huge problem, but there's a lack of definition, and he developed an assay to clarify that lack of diagnosis, and he has been very successful. I think maybe most of you are convinced that he was able to put from the scratch an idea to develop molecular markers, and now he is making diagnosis with this approach. So it's like a whole history in one person, and he has been able to do all this work. In addition, he was able to do this kind of collaboration, as you see in the cartoon. There's a Chilean and U.S. flag, which means that it's two different populations. And I think this is something that is quite important when you are moving to the clinical research, in which you need to revalidate your data in different populations. And he did that, so it's another asset of his successful development.
Okay.
Well, this is more of the same. I think we can skip it.
Okay. Okay, this was a presentation by Veronica Burcio. She is working on the basically the development of therapies based on antisense non-coding mitochondrial RNAs. And so she showed in her presentation that knockdown of such RNAs increased tumor cell death by an apoptotic mechanism, also led to cell cycle arrest, so decreased proliferation, and on top of it, reduced migration and invasion. Essentially, some of the markers they looked at in this study are showed here in the Western blot in the end. So we have, for instance, cell cycle mark, so the cyclins are down-regulated, and as a consequence, basically, we don't have proliferation. And on top of it, she then showed in a large number of different models, mouse models, that for different types of cancers, that knockdown of this particular non-coding mitochondrial RNA is very effective in preventing, for instance, tumor growth, as you can see here on the left, and even things like lung and liver metastasis. So it seems like a very, very promising therapeutic approach that could, of course, be developed in humans to treat different kinds of cancers. And indeed, they've started doing preliminary human trials, and the results so far look very promising. Okay, so on top of the individual talks that we've just highlighted now, we looked for some transversal topics, so topics that were or came up in different talks and that we thought were relevant to what we're talking about here and important to highlight. So the first one is related to the presentation by Manuel Varas, who essentially highlighted again the importance of all these different characteristics that tumor cells acquire. Meanwhile, there are 10 of them. And what this image that shows very eloquently is that essentially exosomes or extracellular vesicles participate. So these are these little bubbles you see here around the circle. Extracellular vesicles, exosomes, participate in the acquisition of all these different traits. So it's really important for the development of cancer, and also has, of course, tremendous potential then, maybe also therapeutic potential, if we can really learn to control these vesicles and what they do and where they go. And indeed, the relevance of vesicles is also apparent in other presentations, for instance, by myself, looking at the role of caveolin-1 in promoting metastatic traits in other cells when present in exosomes that are released, of course, by cells with a higher metastatic potential. And then again in the presentation by Lorena Lobos, showing that lactadorine present in exosomes is important in breast cancer and important for diagnosis, hopefully early diagnosis of breast cancer. Then there was another one relating to what Wael El-Rifai mentioned in his presentation on trefoil factor 1. So he pointed out there that while trefoil factor 1 is very efficient as a tumor suppressor, for instance, in gastric cancer, reducing, so that the presence there of trefoil factor reduces a number of signaling pathways that are important in the development of gastric cancer. But it seems like this is a context-dependent phenomenon, because in other cancers, trefoil factor is actually, expression is enhanced, for instance, in breast cancer, and seems there actually to play an opposite type of role. And so this duality of function of proteins in cancer is becoming increasingly noticeable and important. For instance, also in my talk, I talked about caveolin-1 that can act as a tumor suppressor or promoter metastasis, is what you see in this scheme here. So depending on the presence of certain proteins in a cell like E-cadherin, caveolin will behave nicely and act as a tumor suppressor. But if E-cadherin is lost, down-regulated for some reason, it actually then promotes metastasis, as you see here to the left in the scheme. And Roxana Pincheira in her talk also related to a transcription factor called SAL2, and also to its dual role, let's say, in cancer. So this, of course, is a very complicated issue, because it becomes difficult then to develop treatments based on targeting a specific protein without knowing exactly the context in which this protein will develop a specific function. So we have to know not only a lot about the biology of individual proteins that we can target, but also the context in which they function in one way or another, because otherwise, of course, failure, any kind of treatment will fail. And there are many examples of this. For instance, treatments based on Fas ligand or Fas receptor, which is a death receptor, they failed miserably because people then later on found out that actually there is a subliminal signaling process that is triggered by this receptor that actually promotes tumor development. So it was the approach of using or targeting these types of molecules was doomed to fail because of the lack of that kind of knowledge. Okay, and then basically also we've heard in many talks about the importance of inflammation in tumor development and also progression. So, for instance, in the talk by Wael El Rifai, we saw that trefoil factor 1 blocks interleukin-6 receptor signaling and thereby inhibits central pathway that favors cancer progression. On the other hand, Lisette Layton showed that metastasis promotion by Thy1 in cancer cells is beta-integrin dependent, and in expression of beta-integrin again is dependent on this particular integrin, can be regulated by inflammation or inflammatory processes. Hernan Gonzalez showed that inflammatory genes are useful as genetic classifiers, for instance, for the prediction of thyroid nodules, and that is as to whether to operate or not. And thus, based on these observations, we can conclude that inflammation is a crucial factor in cancer progression metastasis, and also more knowledge about this is important for the development of treatments. And the relevance again of inflammation was also highlighted in the presentations by others such as Denise Bravo, as I mentioned, Marcelo Mosso and Manuel Valenzuela. Alejandro? Alejandro, are you there?
Yes, this is an integration between Dr. Carvajal Carmona, Armensen, and myself, in which, again, we have these novel mutations. But I think here I would like to highlight the point that not novel mutation is driver. That mutation could be found in the whole region, like Latin America, like Nirvana project, or in isolated cases, like the case that I was presenting that session, in which was only one case, but the family contained germline mutations. So sometimes to identify key mutations is tricky, and also as I mentioned, Andrew, the effect of the mutation has to be proved. And with that, I would like to highlight the concept of the clonal or private mutation, because probably if the mutation is clonal, is a driver. But in the case of private mutation, it will be target only one clone of the tumor by itself, not the other clones. So it's quite tricky. And if we are finding novel findings in terms of when we study our own population, we need the capacity. We have to have the capacity first to understand those mutations. So we need a lot of bioinformatics. That's the reason of the session today. And also the capacity to perform functional assays to characterize the biological effect of that mutation. So it's not just simple identifying novel mutations. We have to be able to characterize those mutations from functional studies.
Okay, let me stop the presentation there. Do you want to switch to your presentation?
I sent it already.
You sent it to...
But I can...
yeah, just go ahead and show it yourself.
Okay.
So what we summarized so far are the first 2 days, and then in a record effort, we tried to come up with some highlights of today's sessions, albeit probably somewhat fewer, so we can
have to share.
Mm-hmm.
Okay, this was done today. This is the summarized the presentation this morning about system biology and immuno-oncology. In this talk, this symposium were 4 speakers. One was Elmer Fernandez, who presented data in breast cancer and the integration between several platforms and the correlation with PAM50, which is a standard platform that was developed many years ago. I think it's important here that the capacity that has Elmer and other groups in the region that integrate different platforms and combine novel data that would be useful to understand specific genes that could be exploited as a future target therapy. In this case, associated to DNA damage and cell cycle. And also raise a question that if you target key genes that are like pan-cancer genes, what happens with normal tissues? If those are going to be able to survive when they're targeted. Then the second presentation was Vinicius Maracaja, who is already working in non-coding genomics and trying to identify functional annotations in creating images like this. Let me put it here. Like this one, which I think is kind of think out of the box in terms that all what pathways that we know are regulated by other genes that don't produce proteins, but could be key element in terms of regulation that pathway. One of the issues to highlight the role of this non-coding genomics in cancer is the finding of germinal non-coding mutation that is associated with a familiar form of cancer. So basically what Dr. Maraja is showing here is how to integrate this huge amount of data developed in pipelines to produce that integration. And in this presentation by Dr. Yatzer, I would like to highlight the focus that he presented on non-tumor tissue and the role of immune response in non-tumor tissue. I think it's quite important because most of the people is focusing on cancer, which is shown here, but this non-tumor, non-malignant is not normal. It's probably prepared to develop a tumor, but in a second stage. So probably it's in some ways influenced by all carcinogenic effects. And he mentioned displays this kind of non-exhausted phenotype. And this kind of immune response could be a good prognosis in terms of immunotherapy, which, as we saw also in the Franz Villarroel presentation, not all immunotherapy is successful. And Franz Villarroel presented these wonderful images that I think is fascinating how they're trying to integrate different immune profiles. And this is also, this is not specifically molecular data, it's cellular data. And combine these different profiles, and also he shows some extraction of several tissues, like for example here, that can purify on in situ, have only one specific markers. And among them, there's one marker that's called VISTA, which is very promissory marker in terms of drive the response on immunotherapy. I think this is, yes, this is the last one. There's another one about the presentations of Marcelo Kogan. I don't know if you want to comment those, Andrew.
I can't see them.
Let me put it here. Okay, these are really one on the top of the hour. We didn't have the chance to have pictures here, but I don't know if you want to mention something about this presentation.
Well, I think it was clear from Marcelo's presentation how versatile that these different nanoparticles are and how they can sort of in different formats can be used for different applications, both for treatment of solid tumor growth and also to prevent metastasis. So this would appear to be a very important area of research to worth pursuing in the future.
Okay, this is Nelson Duran.
Well, Nelson basically showed then in animal models they're using first, and then in experiments of sort of preclinical and clinical settings that the nanotherad formulation they have developed is very effective, that essentially has little side effects, at least in dogs. In humans, maybe slightly more, but all treatable, and that these treatments are beneficial to the patients treated. So I think these are also very encouraging studies.
And this is the presentation.
Yeah, I guess I just already summarized that.
Yes, I think the merit of this presentation is an example how to move to clinical data or clinical trials in one specific treatment, which is bladder cancer. This was the second talk of Nelson Duran.
Mm-hmm.
And then Silke Kroll.
Well, I think that was, as usual, Silke, so very innovative, always moving on to new areas, and it was very exciting to see how simply by using different types of surfaces you can encourage different types of cell growth, and obviously in doing so create a great variety of novel sort of models that can be studied in terms of how they respond, for instance, to different kinds of drugs. So it gives us a lot, again, more sort of tools to work with in the lab.
Okay, those are my slides.
Yeah, and I think also the presentation by Marcelo Casanias was rather insightful in that PKCs, what he did there was to, I mean, it's long been known that their activation is somehow or can be related to tumor development. And there's a lot of work on that, but what he showed very elegantly is that the activation of PKCs is also related to activation of pathways that trigger activation of small G proteins like RAC1 and thereby favor processes such as migration, invasion, and metastasis. So that was very nice too. Okay, with that, I think we're sort of coming towards the end here. What I would like to do is to go to share screen mode and present to you the if I can find this in all the presentations I have open here. We had an award for the best image that was actually used in, amongst other, was on the cover page of the abstract booklet that all of you should have received, and I will show you. There we are. So this is the image that we used, and so this is from a biopsy of renal cancer, sort of a triple immunofluorescence image of a histological section, and basically to looking at the distribution of certain proteins, fibronectin, tenacin, in the tumor stroma. And of course, in blue, you also see the nuclei of different cells. And this, the prize was awarded to Jenny Enriquez. Was selected from about 10 different images that were sent in. And this is Jenny, and she will get her prize in due time. It's basically an acknowledgement of her participation and contribution to the meeting. And finally, I would like to move and finish off here by acknowledging the participation of a number of people. Who helped us make this work. If I can just find this thing here somewhere. There we are. And so like to particularly acknowledge the contribution of Maria Jose Olivares and Adriana Mujica. They have been operating, of course, behind the scenes. They basically made all of this the possible. They developed the abstract book. They organized all the links, had us also on YouTube and so on. So this meeting was heard and followed by a considerable number of people beyond those present at the meeting, in the Zoom meeting. So great work there. Then I'd also like to mention the contribution of Ricardo Wilcoman and Andrea Rodriguez, who did an excellent job in putting together the video that you've been seeing in the coffee breaks, summarizing the previous meeting in 2016, and also events at the recent meetings, so November 3rd, 10th and 17th. Then also the participation of Lisette and Marcelo Hermoso in generating the highlights, particularly of the first 2 days, the 3rd and the 10th. And then, of course, I'll acknowledge the participation, enormous contribution of my co-organizer here, Alejandro Cordovalan. I think it's been really great the way we put this together. There was a strong participation of basic science, but also together complemented by a very strong participation in clinical and translational research. And so I think this really made this a very interesting meeting. And with that, I would just like to thank you all for your participation here. I hope it's been a stimulating meeting and will have generated some new ideas for future research and also particularly collaborations that might emerge. It's also an objective of these kind of meetings. And I think to finish off, just like to tell you, hope to see you again in a future meeting. We are planning on organizing another one in a couple of years, 2-3 years, and we do hope this time it will be presential again, so not just virtual. We'll actually be able to see one another and talk to one another directly face to face. Would be nice once we get over the COVID-19 crisis. And to finish off, I believe Maria Jose has put together another little video. And so just to say goodbye then, Maria Jose, are you ready? Está listo con el video? I don't know.
Please wait 1 moment.
Okay.
It's coming, it's coming.
So anyway, meanwhile, goodbye to everybody. And as I said, we hope to see you again in the future.
Next council meeting.
Obviously.
See you.